Excellent. So we're now being recorded. So today is Monday, April 27th. We are doing our last lecture of the digestive system. There's a couple things that are going to be due on Wednesday, obviously. The first is that there's the extra credit histology assignment. We talked about the histology thing in class on Wednesday, and then as I mentioned on Friday, in talking with my 4:30 class on Thursday, it seemed like such a good idea that we were doing it here that they should do it as well. I should take advantage of that with them as well. And in talking with them, because it was outside of normal class time and there were issues that way, and we had talked in this class about doing extra credit, we decided to do the drawings, kind of like way back in 4:30 where you had to do the epithelial tissue and connective tissue drawings for the histology to encourage you to actually spend the time doing this material. The extra credit is for doing that. So because I didn't get a chance to specifically talk about that assignment in class, I did make it due today. I don't normally like having assignments due on the day of the exam, but rather than having this, in case nobody bothered to listen to the histology review, I wanted to make sure that this was during the official time where we talked about it. So again, you are responsible for, in your digestive system study guide, the last, I think, 4 pages of it are 3 or 4 pages are histology. Those histology drawings, all of the drawings, and labels. I don't want lines drawn. I don't want numbers written next to them. I want them labeled properly. Those drawings are due by 7 o'clock on Wednesday morning. So that gives you plenty of time. If you haven't done it already, first and foremost, shame on you. But secondly, please get that done. Again, color is not required, but it is appreciated. So anything that helps you to be successful in studying this material. But it is a great way to learn that. Again, use the resources that you have, your histology atlas, your textbook, your lab manual, the professional anatomy lab that's on Mastering A&P. There's some great histology sites, as I talked about during the histology thing. Like a third of the pictures easily came from a Yale histology site that I found, which was excellent. So there are a tremendous number of great resources, as well as the YouTube histology lab that we did to help you to complete that activity, and that is due on Wednesday. More important than that, and that is extra credit. That will be for 10 points of extra credit. So yeah, let's write that. So 10 points of extra credit for completing that and turning that in. So definitely you want to do that. Obviously, the most important thing that is going to be going on Wednesday is we are having the lab and lecture exam. Again, there's a lot of similarities to the format of what we did last time. Again, the exams will be available from 7 a.m. To 2 p.m. And that is when they're available. And they also must be completed within that time. If you wait till 1:50 to start one of the exams, then you're only going to have 10 minutes to complete it. The format is the same as last time, in that both exams will be available during that entire time, and you can take them in either order that you want. The format for the lecture exam is going to basically be identical to the previous. So what that means is that it's going to be, no, there weren't extra credit questions on the last exam, and so there won't be in this one as well. So this is the format for the lecture exam is going to be identical to what we did for lecture exam 3. So again, there's going to be multiple choice questions, fill in the blank, and essay questions, all randomly assigned. And again, all questions will be presented at once so that you see the whole test in front of you, and you can answer it in any in any order that you want, and then you just submit it when you are done. So again, there is nothing new about how the format of the lecture exam is going to be. I haven't finished writing it, but my guess is that it'll, again, somewhere be around 2.5 hours to complete it, because it'll probably be about a similar number of questions, maybe a little less. So maybe if it's a smaller test, it may be a little less time, but my guess is somewhere between, so let's be more specific, between 2 and 2.5 hours, depending on the size of the exam. What will be different is going to be the lab exam. There is going to be a similar number of questions on this lab exam. It's going to be 65 questions. and you will have 75 minutes to complete the exam. Most people didn't run into a time issue on that, so that should not be a problem that way. However, what will be different about this lab exam is in looking at it and in sampling it and trying it out, I think I am going to change the format. So the format for this will be the same, not similar, but let's be more specific, same as the lecture. So what's going to happen is all the questions are going to be presented at once. It is a lot with 65 questions to have to scroll up and down. So you still have to be careful of your time. You know, start at the top and work your way down. But if you see something to question, 25 that makes you question what you wrote at your answer at number 2, you do have the opportunity to scroll back up and see it. So all of the images will be all on there, but they're big images, right? There's the space to write, which is big. So it is a long test. So try not to scroll around too much on that. And then again, you'll answer all the questions. And if you're jumping around, make sure that you answer all the questions. That's the other fear of this as well. If you're jumping from question to question to question as you're doing that, you may skip a question. So make sure you are thorough, make sure you are complete in that. But like I said, there were not many problems on the last exam between the two classes, but those few the problems that did come up came up in the lab exam in that format of one question at a time, and loading speeds and times and things along those lines. So hopefully this format will work better. So hopefully there won't be as many problems with this. Hopefully it'll do it okay. Hopefully you won't then have as many problems. It may take longer for it to populate at first, but then once all the images and everything populate there, it should be pretty simple and easy to go through. So it should be hopefully simpler, hopefully take even less time than it did before, because you can, pace yourself, but be comfortable that you can go back. So hopefully that will be successful. So that is going to be the format for that as well. All right. Any questions on any of that? All right, and then when we're done with this, I'll tell you right now, just like last time, we are going to, after this, dive straight into the reproductive system. The reproductive system is one organ system, but it's really two, because obviously the male and female are dramatically different from each other, but obviously intimately related. So in all likelihood, and I don't remember off the top of my head which one it is going to be, but there will be a pre-lab that will be due on Monday when we come back. So that way it encourages you to start looking at the material ahead of time. But what I'll tell you right now is we've got the exact same format for the reproductive system that we did for the digestive system. We have 3 lectures and then an exam. So it's another quick 2-week turnaround for one organ system, and this big organ system, really 2 organ systems. And then after that, we have the final. So we are rapidly approaching the end of the class. And so that'll be big as well. And so that'll be the other thing that not only will I have a pre-lab due, but I also have a review for the final that I'll be posting as well. So again, not something you necessarily need to be looking at yet, but I will post that so that you have the opportunity to look at that as well. All righty. I think that is the game plan. If there aren't any questions on that, I think it's pretty simple and straightforward. No new information on that. So last chance for questions. All right, then let's switch gears and head back into our lecture. And then I can go ahead and turn this off so you can stop staring at me and my glorious beard, and we can move on. All right. We left off last time. We had just finished talking about the small intestine, the primary organ of the alimentary canal. And what we were talking about, getting ready to talk about, were the accessory structures. Some of these are things that we have already talked about, so there is some familiarity with that. And one of those is definitely the pancreas. The pancreas is a feather-shaped organ. And remember, as we talked about, the body of it and the head of it are retroperitoneal. Right? If you think about it, so is the duodenum is retroperitoneal, and as you can see, the head of the pancreas basically is wrapped around by the small intestine. The duodenum of the small intestine, which is also retroperitoneal, wraps around the head of the pancreas. But interestingly, and I have no reason, an idea why, the tail does stick out into the abdominal pelvic cavity, so it is intraperitoneal. The pancreas and the liver together are two major accessory structures that help to produce secretions that release into the duodenum. As you can see here, this picture shows it really nicely. There are actually two openings to the pancreas. The largest duct is the main pancreatic duct. It runs the entire length of the pancreas and then expresses into the duodenum. Notice it's easy to identify the main pancreatic duct because it is met up with the common bile duct coming out of the liver and the gallbladder, and we'll talk about that anatomy a little bit later. Notice there is an additional second, what is known as accessory duct to the pancreas that also releases into the duodenum by itself. As we talked about both in histology and way back in the endocrine system, there are two components to the pancreas. It is a mixed gland with two different types of secretory structures. We have the acini, which produce the pancreatic juice. They are exocrine in function. And we have the islets, which are endocrine in function, producing, 4 different hormones with all of the cells like insulin and glucagon that we find in there. So as I mentioned, here we see again, that pretty picture of that main pancreatic duct and then the accessory. And again, we can tell the main because it meets up with the common bile duct of the liver and gallbladder. There is an enlarged finger-like projection that sticks out into the duodenum where the pancreas and the liver and gallbladder express their secretions, known as the duodenal papillae. So we have that papillae there, and that's another way we can tell the difference between the accessory and the main pancreatic ducts is the location of that. We already talked about the acini being exocrine in function, and here we see them feeding into those ducts that would lead to the main pancreatic duct. And again, remember, notice they've conveniently put a whole lot of blood vessels around our islet to remind us that it releases its contents into the interstitial fluid and is endocrine in function. As I just briefly mentioned and want to show again here in this nice illustration, as I mentioned, the common bile duct from the liver and gallbladder and the main pancreatic duct come to the same release point. As we see here, there is that enlarged projection that sticks out into the duodenum called the duodenal papillae. But if you notice also, there is a big, large open space here known as the duodenal ampulla. And the release of these secretions are controlled, as you can see, by an enlargement of the circular muscle layer. Notice the circular muscle layer wraps around these two ducts where they come together, forming a large valve-like structure known as the hepatopancreatic sphincter. Hepato, of course, means liver. Pancreatic, of course, refers to the pancreas. If you remember, this hepatopancreatic sphincter is controlled by cholecystokinin. Oh, why is that so teeny? Maybe that, I don't like that. Let's go a little bigger. Is controlled by cholecystokinin. So cholecystokinin, remember, is a hormone that relaxes that hepatopancreatic sphincter, allowing those secretions, allowing that bile, allowing those enzymes to be released into the duodenum to help to break down the chyme. And of course, remember, it is lipids that is the stimulus for that, and we'll see that when we talk about the function of the liver as well. All righty. And clear that. Again, depending on how often you eat, the composition of the food you eat, the volume of the food you eat, it can vary dramatically, but in general, our pancreas produces about 1.5 liters of pancreatic juice during the course of the day. That pancreatic juice contains enzymes, water, ions, and bicarbonate. Again, remember, we have not just bicarbonate, but also other phosphate buffers as well. Not surprisingly, our pancreatic juice is fairly basic, with a pH of about 7.5 to 8.8 on the pH scale, because remember, this is released into the duodenum, which is what receives the chyme directly from the stomach. And of course, the chyme, as we've talked about, has been in the stomach, so its pH is around 1 to 2. So these buffers are going to play an important role in helping to neutralize the acidity of that chyme. Remember, our stomach has all sorts of protections and that big, thick, sticky alkaline mucus to protect it and buffers it, but it also keeps the chyme away from the wall of the stomach, protecting it that way. We don't want to keep the chyme away from the wall of the small intestine. The small intestine contains important enzymes and is where the absorption is going to take place. So we need to neutralize it so it's not damaging the lining of our small intestine. And as we talked about, the release of our pancreatic juice is controlled, not surprisingly, by both neurons and by our endocrine system. Both are sympathetic and parasympathetic. Parasympathetic obviously increases the production and release. Sympathetic decreases production and release. And remember also way back in that cephalic phase when we were talking about regulation of the stomach, remember we were focusing on the stomach, but we also said that those neural controls would affect other things as well. And so during that cephalic stage, while our parasympathetic nervous system is priming the pump of the stomach, it also is going to stimulate the pancreas to start producing juice to be able to be released as well. And like I said, the expression of that those secretions into the duodenum is done primarily by cholecystokinin, which is that important hormone that is going to release the hepatopancreatic sphincter. Now, as I mentioned, the small intestine is the rock star of the digestive system. This is where a majority of the chemical breakdown and the majority of the absorption takes place. And while the small intestine is the location, it is primarily the pancreas that is responsible for producing most of the major enzymes that are going to play a role in the breakdown of our macromolecules. So our pancreas produces a massive amount of enzymes. Some of the major enzymes, one of the most common enzymes produced by our pancreas is trypsin, but also along with that are chymotrypsin and carboxypeptidase. Anyone have any idea what trypsin breaks down? Trypsin, you need to know these enzymes and what their functions are. So what is trypsin responsible for breaking the bonds of?
Carbohydrates.
Carbohydrates? Nope, try again.
Proteins.
Proteins, there you go, absolutely. Proteins, right. You're right, we do want to break down carbohydrates, but for that, we are again going to be using an amylase. This amylase is produced by the pancreas, so it's pancreatic amylase. What do you think pancreatic lipase breaks down?
Lipids, fats.
Yeah, pretty simple, fats. And here's another one that's pretty easy as well. Ribonuclease and deoxyribonuclease. What do you think they break down?
Nucleic acids.
Amino acids.
Yeah, there you go, our nucleic acids, right? Remember, if you think about it, when we talked about these things, our carbohydrates, that breakdown started, remember, in the mouth. For our proteins and for our our lipids, this starts, so proteins, remember that start in the stomach with the pepsin, lipase, our lipids, they started also in the stomach. Right, that one's a little bit of a cheat because remember it's our tongue that produces one of the enzymes, but remember it's not active to the stomach. But here with our nucleic acids, right, these actually start and finish here in the small intestine. I'll just put SI because I'm running out of room. There you go, excellent. So we have all these enzymes to, in some cases, start, and in all cases, complete the chemical breakdown of our macromolecules. Now, if you remember in the stomach, we talked about how pepsin was produced in an inactive state, pepsinogen, and then it was activated once it was released from the cell. In the case of the pepsin, pepsinogen is activated by the hydrochloric acid produced by the parietal cells and other digestive enzymes in there. Other pepsin that's in there can activate more pepsin. We have a similar thing here where our enzymes are going to be released in an inactive state. However, while as I mentioned, the pancreas produces a majority of the enzymes, they don't produce all of the enzymes. As I mentioned before, but it's been a while now, on the surface of the absorptive cells, so those simple columnar cells that have their microvilli, right? So remember, those absorptive cells are simple columnar with microvilli. On the surface of those absorptive cells, embedded within those microvilli, are many, many enzymes, all sorts of different types. These enzymes you don't need to know the individual names of. The ones up here listed above, you do need to know the individual names and what they do. But in the case of the small intestine, collectively, they are known as brush border, because if you think about it, the microvilli kind of have a bristle-like appearance to them. They're called brush border enzymes. And these, oops, did I spell enzymes right? These brush border enzymes basically do two things. They play an important role in the activation of these pancreatic enzymes. But the other thing they are often doing is completing the chemical breakdown. So they activate pancreatic enzymes and they complete the chemical breakdown of our macromolecules. So when we talk about the absorptive pathways, these brush border enzymes are going to play an important role in that as well. Your book has a pretty picture that shows this. Like I said, I'm not going to hold you responsible for the specific pathway of this particular one. Let's go ahead and clear that so that we can go here. Here we see an example. Notice, for instance, our pancreas is releasing that trypsinogen. Yeah, primarily it's one of the main reasons for it is so that the pancreatic enzymes don't damage the pancreas itself, absolutely. And so that they're only activated when they're needed, right? When they're being released into here, and that's when they're being needed. So notice again, you don't need to know this pathway, but notice here's that trypsinogen, which is the inactive form of trypsin. There is that, and notice it's an enteropeptidase, which again is not a name you need to know, but these brush border enzymes on the microvilli of those cells that activates the trypsinogen into trypsin. And notice also, most enzymes, not most enzymes, all enzymes are proteins. So not surprisingly, the enzyme that plays a role in breaking down proteins can also play a role in activating our other enzymes as well. So again, your book shows a nice pretty picture. I wanted to show you this. You don't need to know any of this pathway, but know the important function of those brush border enzymes. All right, that is our pancreas. So what we need to focus on now is our liver. Our liver, we could really literally spend the rest of this class, the entire class, talking about the function of the liver. So we are going to paint with some really broad strokes. We talked about this a little bit in our histology, a lot of bit in our histology. And also, I made a point while I was working on the lab exam, like I said before, that CRC site has some great images, both models, charts of the classroom. I guarantee you those charts are going to be of the type of materials that would be on the exam and the models and things along those lines. But what I found is they were lacking in two major areas, two of the models that I absolutely love in the classroom. And if it's something that I love in the classroom, you know it's going to be on the exam. So I made them those images available under the screenshots for you under the module, and we'll talk about them briefly as well. But let's talk a little bit about the anatomy of the liver, starting first with the gross anatomy. The liver is the largest visceral organ takes up basically the entire upper right-hand quadrant of the abdominal pelvic cavity. It is somewhat asymmetrical in its shape, though. The liver is made up of 4 different lobes. By far, the largest of those lobes is the right lobe. Here we are looking at a liver from an anterior view. This is an anterior view of the liver. Here is the right lobe, here is the left lobe, and you'll notice that the lobes are divided by a thick connective tissue fascia, and this thick connective tissue fascia is known as the falciform ligament. Notice I did not extend that ligament all the way down out of the liver. There is a connective tissue structure that looks continuous with the falciform ligament, but really isn't truly. And as such, we give it a different name. This structure, and let's go ahead and change the color of this. Oh, red's too similar. We'll go green. This structure that extends out beyond the liver is what is known as the ligamentum teres, or the round ligament. If you remember in the heart had some vestigial structures. The fossa ovalis, the ligamentaris arteriosum, that were left over from embryonic development. And that's actually what the ligamentum teres is. As we will talk about when we get to the reproductive system, as most of you know, when you are attached to mom, you are done, that is by an umbilical cord, so that mom can provide the oxygen, mom can provide the nutrients, and at birth, that is cut and basically shrivels up and becomes your belly button. Well, the same way that we have a special hepatic portal blood vessel system, where as we get our nutrients from the digestive system, it goes first to the liver, it's the same thing for baby as well. Those blood vessels coming in through the umbilical cord actually go first to the liver, right? Because mom's not always eating necessarily the most nutritious things, the most well-balanced things. So again, it may be nutrient rich, but it isn't nutrient appropriate. And so those blood vessels pass through the umbilical cord straight to the liver. Well, when you cut the umbilical cord, it shrivels up on the inside as well. And that's basically what the ligamentum teres, that round ligament, is. It's the vestigial blood vessels that passed through the umbilical cord on the inside. So our liver has a belly button as well. This is the anterior view. If we look at the posterior view, there's a couple interesting things we can see. One of the cool things that we see is, notice there's this big, huge indentation in the posterior side of the liver. As you can see, it's got a fancy name. It is the sulcus for the inferior vena cava. Sulcus, remember, we just know, means a groove or an indentation. But basically, as it indicates, this is where the inferior vena cava sits. So basically, the liver wraps around the inferior vena cava. And again, there are 4 lobes, as we mentioned. We have that really big right lobe. We have the really big, well, not really big, but relatively large, but still much smaller than the right, left lobe. But notice right up here superiorly and right down here inferiorly, there are 2 more lobes. They are the caudate lobe and the quadrate lobe. And if you have trouble remembering which and which, I just remember they're in alphabetical order. Caudate is superior, quadrate is inferior. So those are our 4 lobes of our liver. Our liver is divided up into individual lobules. These lobules are the structural and functional units. And it is, like I said, the largest visceral organ. These are hexagon shape, and we talked about this a little bit in the histology. They're very nice and distinct in their structure. They are hexagonal in shape. There is a large blood vessel at the center called the central artery, central vein. And then we have all of the liver cells kind of lined up in these rows. And of course, the liver cells are the hepatocytes, lined up in these rows from this radiating outward. As we talked about, and we'll see in the illustration, we have that hepatic portal triad, those three structures that are located in all of the corners. So there's going to be one there and a hepatic portal triad there, and so forth and so on at all of the corners of those. And then the central vein at the center of it. Right, so we have all of this anatomy. We talked about that a little bit during the histology. I'm not going to go over it too much because you can go back and you can read that, but we will look at it briefly because there are a couple important things we need to emphasize and see. The cells of the liver are called hepatocytes, and they are true workhorses in the body. Their job, as we talked about, is to maintain appropriate nutrient levels in the blood. That means things go both ways. If there is too much glucose in our blood, then it will absorb glucose and store it in the form of glycogen. If there is not enough glucose, it will break down that glycogen and release it, right? If there's not enough manganese, it'll release it. If there's too much, it will store it. So both its job is to store excess and replenish when too low. Liver cells are also responsible for detoxifying the blood. They play an important role of removing any toxins, right? If you haven't washed the pesticides off of your foods properly before you ingested them, it can help to break those down. But again, toxins, we can kind of put that into quotes. Toxins can mean all sorts of things. It can also mean the medications that you have ingested. It can also mean the hormones that your body is producing, things along those lines. It is going to remove all of those types of materials. In fact, how good your liver is at breaking down a medication determines the form of which you take that medication, right? Some medications can be swallowed in a pill, because it'll be able to, doesn't get broken down as quickly in the liver, so it can pass through the liver on the way. Because remember, anything you ingest is going to pass through the liver first. However, if the liver is very efficient at breaking down a medication, then instead you have to either take it sublingually or you take it in an injection or something like that, where it isn't all going to the liver first. Right, as we talked about, our lactic acid from our workouts gets broken down in the liver. And like I said, those hormones which we produce, those long-lasting hormones that get circulated through the body, are going to get broken down by the liver as well. If that's not enough, our liver also plays an important role in the production of bile. Bile plays an important role in the emulsification of fat. Again, when we are emulsifying, that is a mechanical breakdown, right? As part of this, again, you did the PhysioX, so hopefully this makes some semblance of sense to you. But if you didn't, or well, first and foremost, shame on you. But another simple way you can experience that is to think of it back to, this weekend, Saturday night was lasagna night. So you make your lasagna as you always do. And after you get done with that, you fill that lasagna pan with water so that it can soak, so get all the baked crust off of it. And there's, of course, that huge, beautiful pool of fat that forms in the middle of it all. The oil forms that big, huge oil spill in the center. And if you just take a single drop of detergent and put it into that water, that whole pool of fat goes away. It doesn't break down the fats. They're not being broken down into the alcohols and the fatty acids, but it does basically take that one big globular structure and break it down into a bunch of small structures. And that's what that emulsification is. It's like we talked about. If you have that boulder and a hammer and a chisel, you use that hammer and chisel, you can break that one big rock into a whole bunch of tiny little pebbles without changing the chemical composition of it. And that is what bile does. The other important thing that is found inside the liver, really in the capillaries of the liver, are our Kupffer cells. Kupffer cells are phagocytes. And remember, phagocytes can act as antigen-presenting cells. However, unlike other phagocytes that we've talked about that migrate through the blood and in fact can leave the blood vessels and migrate through the tissues, these Kupffer cells are actually embedded in the walls of the capillary, so they do not move. They do not leave the liver. But again, remember, all the food you ingest, all of that goes into the blood, and all the blood from the digestive system goes to the liver first. So a lot of things that are entering in the body have to pass through the liver first. So having a bunch of phagocytes there to check what's going on and what's coming into the body is something that is incredibly useful. If they come in contact with a harmful pathogen, they can break down that pathogen and present its antigens to help to stimulate an immune response. These Kupffer cells also play a role in helping to store, I think I say it right here, so I won't say it. Yep, there we go. Plays an important role in storing iron and other heavy metals, as well as lipid-soluble vitamins. So it's an important storage cell in that way as well, so they can play a role in that as well.
Those are the Kupffer cells specifically?
Yes, Kupffer cells specifically, yes. All right, so as you can tell, there is a whole lot going on in the liver, and here is the pretty picture that we did. We talked a little bit about and looked at histologically and the things that I just drew. So notice this would be an example of one of those liver lobules. It has this hexagon shape to it with the lines of hepatocytes running through that way. And at the center is that central vein, as we talked about. One of the other things we talked about, and I think for this I want to switch. So let's go back real fast before we get too deep into this. Let's take a look at one of those pretty pictures that I was talking about. That I posted on let's see, that I posted on your Canvas site. So, as we talked about during the histology, and I think we talked about it a little bit, there are basically two blood supplies to the liver. All right, so let's move this over here. Can you guys see all of that?
Yes.
Okay.
Yeah.
So, two blood supplies going to the liver, and let's go make sure to emphasize that. Going to the liver. The first, as we keep talking about, is the hepatic portal system. These are veins from the digestive system. They are, of course, systemic veins. So these are oxygen poor. Right? After all, they just carried their blood to the digestive system. As we'll see, most absorption is an active process in the small intestine, requiring energy, requiring oxygen. So it is going to be oxygen poor, but because it is coming from the digestive system, it is nutrient rich. And this is the blood we need to be processed. We said it's nutrient rich, but it needs to become nutrient appropriate. The second blood supply going to the liver is the hepatic artery. This is a systemic artery. And so not surprisingly, it is oxygen rich, right? And let's go back. Systemic artery, it comes off the celiac trunk, right, and the common hepatic artery, which also feeds the right side of the stomach, right? Hopefully you remember that pathway, and it's oxygen rich. So we have these two blood supplies coming into it. And if you notice, this is a kind of posterior, inferior, kind of weirdish kind of view. It's mostly an inferior view of the liver, but they've cheated a little bit on it. So for instance, if we would look closely, we can see this part over here is, we can't appreciate it and see it well, but this is the larger right lobe, and this is the left lobe. However, here we have the caudate and the quadrate. So they have shown all four lobes for us on that. But that's not what I like about this. What I like about this are all of these vessels that we can see in here. We see blue things, and blue things we know definitely means veins. red things, we know red things definitely mean arteries. Okay, not necessarily in the pulmonary system, but we're not in the respiratory system. So it's usually a safe guess that red means artery and blue means vein, or at least it means oxygen rich and oxygen poor, which is what we expect here. And as we have already mentioned, hold on, I need to move this out of my way. The liver wraps around the inferior vena cava. So here, notice we have that inferior vena cava that the liver is wrapping around. Now, as I mentioned, we have two blood supplies coming in to the liver, and the first one starts right here. So this nice blood vessel that you see right here is our hepatic artery. Right? And that's an artery, and that's a vein, and this is blue. But there's this weird gray looking blood vessel. And this gray looking blood vessel they've used to represent our hepatic portal vein. Right? It's not red because obviously, as we know, it is oxygen poor, but it is going to be nutrient rich. And so these are our two blood supplies coming into our liver. We bring the oxygen-rich blood so that we have the oxygen for the liver to do work, and the blood from the digestive system that needs to be processed. And then down here at the microscopic level, down in here is where all of that processing is going to occur. Once that processing is occur, we have two blood supplies that go into the liver. We have one blood supply that comes out. And that one blood supply that comes out has been processed. So this means it is going to be oxygen poor and nutrient appropriate. And again, if you remember when we talked about our cardiovascular system, the blood vessel that comes out of the liver is the hepatic vein. Notice, not hepatic portal vein, right? So this is the hepatic portal vein. This is the hepatic vein. And it's the hepatic vein that feeds into the inferior vena cava. So notice, if you see, there is a third type of blood vessel here. And that third type of blood vessel are these blue blood vessels, like that one there. And even better, although, hold on, I need to cheat and move these things. Let's move that there. And then this can be moved there. And then I can more clearly and more specifically point at this right here. Notice this blood vessel here feeding directly into the inferior vena cava. That is, of course, our hepatic vein. Right? Not portal vein. Hepatic vein and hepatic portal vein do not mean the same thing. Hepatic portal vein is what feeds into the liver. Hepatic vein is what feeds out. And this does a great job of showing that. Notice, since we're here, the other cool thing that this shows, and ironically, I've already been using green, so I'll change colors now. Let's go with, we'll go with what'll stand out. Purple maybe will stand out. That's pretty good. Notice the other function, as we talked about, for our liver is that it is producing bile. That bile needs to leave the liver. So there's things coming into the liver and things that need to come out of the liver. And that is going to go out to the duodenum. So notice here we have two ducts that are feeding out of the liver. So this is the, remember the side's the left because we're looking at an inferior view, the left hepatic duct. And then this one over here is the right hepatic duct. These feed into a single duct for both of them. So sneak this in this way. Oops, that's not going to work. Do that the other way around. The left and right feed into the common hepatic duct. Notice hepatic ducts because these are all related to the liver. And then that feeds into this cut duct right here. And this cut duct right here is our common bile duct, because this contains bile from both the liver and the gallbladder. If you have ingested a large amount of lipids, then what happens is your liver is going to be producing that bile, releasing that bile into these hepatic ducts, into the common bile duct, and that will be expressed into the duodenum through the hepatopancreatic sphincter. However, your liver is constantly making bile, and hopefully, even though you're quarantined at home, you are not constantly eating fat. So what will happen in those instances is that rather than being expressed out the common bile duct, what will happen instead is that, that bile will move up a duct known as the cystic duct. And we're going to put an asterisk in front of this one. Oops, in front. The cystic duct here to our gallbladder. That gallbladder's job is to do two things. It is going to store and concentrate bile. Like I said, your liver produces bile, but your gallbladder pulls water out of it, concentrating it. In fact, the bile in your gallbladder can be as much as 10 times as concentrated as when it was first produced, making it far more potent. Right, so that way when we release the bile from it into the small intestine, it can be really, really efficient at breaking down that fat. However, remember, its goal is not to make it. So if you get gallstones or your gallbladder is injured and has to be removed, you can still eat fats. Your liver will still produce them. And in fact, these hepatic ducts will actually swell slightly to be able to store a little bit more of the bile in there. But notice the liver won't be able to concentrate it. So you'll be able to release bile into the small intestine, but it won't be as concentrated and it won't be as efficient at breaking down fats. So if you lose your gallbladder, you don't have to stop eating fats, but you may have to switch from a 3-scoop ice cream sundae to a 1-scoop ice cream sundae because your bile won't be as efficient at breaking down those fats. All right. Notice the reason we put a asterisk next to the cystic duct is this cystic duct is the only duct where bile can flow in both directions. Right? If you think about it, when we produce the bile, bile is always coming out the left and right hepatic, always coming out the common, always coming out the common bile duct. But depending on the situation, it is able to go up the cystic duct to the gallbladder, and then also, when it's time, come out the cystic duct to be expressed. So the cystic duct is the only two-way bile duct. All the other bile ducts, all the other ducts that carry bile, I should say, only do it in one direction. All right, so this is an awesome model. I guarantee it's going to be on the exam, so make sure you are familiar with that. All right, the other thing that I wanted to show you, and let's come back to this pretty picture. As we come back to, actually, I guess I should go back, although I don't know if it's going to still be there. Let's find out. Nope, all the pictures are gone. Oh, well. You've got it, and you can always freeze the screen during the lecture if you need help on that. All right. The other thing that I wanted to talk about for this, and now we look at this at the microscopic view, notice we can see those structures that we were just talking about. Those large blood vessels that we see feed into this come to our liver lobule over here. So notice here, as part of our portal triad, we have a branch of the hepatic artery, bringing the oxygen-rich blood. We have a branch of the hepatic portal vein, bringing the nutrient-rich blood. And notice they both feed them into these large capillaries that penetrate from the edge towards the center. These are big, large, loose, incomplete lumen capillaries. So I'm sure it says it somewhere here on this picture, but I'm not going to bother trying to find it. These are our sinusoid. Oh, it does say it down there at the bottom. These are our sinusoid capillaries. Remember, we talked about the liver was one of the places where large things were able to get in and out. And notice the hepatocytes line these sinusoid capillaries. And, oh, look at that. Right here in purple, we have one of those Kupffer cells. There's another one right there. Those Kupffer cells embedded within the wall of those capillaries. So if you notice, and I'm going to increase the size of it here, and an arrow, and make it red. Both our blood, our oxygen-rich and our nutrient-rich blood, is traveling from the periphery of the lobule towards the center. So again, we have oxygen-rich blood in the artery branch. We have nutrient-rich blood in the hepatic portal vein branch, feeding into these sinusoid capillaries and bringing it to the center where that central vein is located. That central vein then it collects the blood that has now been processed and is oxygen poor and nutrient appropriate, and it sends it out, as you can see, to the hepatic vein. But at the same time, all these hepatocytes are also making bile. And as these hepatocytes are making bile, they're releasing that bile into these tiny little bile ducts, right? They're like tiny little canals, and so not surprisingly, these tiny little canals are called the bile canaliculi. And those bile canaliculi collect the bile and bring it out the liver lobule to the bile duct. So notice blood goes towards the center, bile goes away from the center into our bile ducts, and then is expressed out into the common hepatic duct, and I mean into the left or right hepatic ducts, into the common, and so on and so forth. In that path we go. So here we see this great three-dimensional image of this. And again, your book's got some great images that show this. So let's take a look at some more of these. Here we see them with the blood vessels. Here we see they've removed the blood vessels. So again, we can really see those hepatocytes. The bile canaliculi are these green spots carrying them out. We see that nicely here. So again, bile comes from the center out. Blood goes from the edges in. And so notice again, there is a connective tissue septum between the individual lobules. And within those septums at the corners is where we have these three structures, which collectively are known as the hepatic portal triad. Bile duct, also the bile channel. Sure, yeah, you can call it bile channel. I would be okay with that as well. Absolutely. All right. Oh, look, here we get some great example of those Kupffer cells. So again, we see that nice structures, the hepatocytes, the central vein, our Kupffer cells in those sinusoid capillaries, hepatic portal vein, hepatic artery, carrying the blood in, and then the bile canaliculi carrying it out to the bile duct for that portal triad. All right. So that is all the anatomy that we were just talking about for this. And we see it really great in these nice, beautiful three-dimensional images. All right. So again, blood enters from the hepatic artery and the hepatic portal vein from the triad, flows through, and again, hepatic artery contains oxygen, hepatic portal vein contains the nutrients that need to be processed, flow through the sinusoid capillaries, into the central vein, and that central vein feeds into the hepatic vein feeds into the inferior vena cava, and the inferior vena cava feeds into the right atrium of the heart. Again, remember these hepatocytes, it's two-way. If there's excess nutrients, they will absorb it. If there is deficient in nutrients, if it has it, it'll release it. And that's the issue, right? If you skip one meal, your liver's got plenty of things stored up. if you skip 17 meals, right, there's only so much glucose that a liver cell can store. There's only so much, you know, magnesium or things along those lines that can be stored. So prolonged malnutrition, your hepatocytes are not going to have sufficient resources to be able to secrete those to maintain appropriate levels. And like I said, if those hepatocytes weren't busy enough, they're also producing bile as well, collected by the bile canaliculi and flowing in the opposite direction. So like I said, we have all these great pictures that do a really nice job of showing this. But the other place where we have it, where it is super, super cool, or at least I think it's cool, and I guarantee is going to be on the exam, is this beast right here. Right. I love this model for many reasons. One, because it weighs about 75 pounds. So when you're in the classroom, you actually get to learn and exercise at the same time, because this thing is massive and super heavy. I'm pretty sure it's made out of, I don't know, whatever something really dense is, concrete blocks or something like that. It is massively, massively huge and heavy, but it is really, really useful as well. Notice basically what they've done here, and let's cheat and go back an image. Go back 2 inches, is what they've done. Notice how they've cut a wedge out of our lobule here. Well, instead, what they've done is basically, and let me think of how it's lined, perfect. What they have done is taken a wedge out of this lobule, and they've done it in this fashion. So they started at the central vein here on the left and went over to the triad on that side. So if you notice, when we look at our picture, lo and behold, this over here, oops, not that, that, I guess I have to label it here. That's too big now, I don't like that. All right, that now is our central vein, and that would be the center. This would be the edge of our liver lobule, and not surprisingly, as you can see here, we have our hepatic portal triad. Right, we have a red vessel, and of course, what is that red vessel?
Hepatic artery.
There you go. It's a branch of the hepatic artery. We have a blue vessel. What is that?
Hepatic portal vein.
There you go. We have a green vessel. What, of course, is that?
Bile duct.
All right, our bile duct. And of course, like all triads, we have a fourth structure, a yellow structure. Wait, what? What the hell is that yellow structure?
Lymph.
Not a bad guess. That is a really, really good guess, but it's not. But I'm talking about it, so what is it much more likely to be? Being that I'm a neuroanatomist and I love nervous structures, what do you think it is?
A nerve.
It's a nerve. Yeah, exactly. So it's a nerve. So it's not really, that nerve is not really a part of the triad. Notice this model has it. It is, in fact, going to be there. Nobody normally draws it, but the model has it there, which is another reason why I love this model. Notice we then clearly have our beautiful hepatocytes. That we can see very clearly. We have our bile canaliculi, right? I think I've got, oh, that's what was wrong. That feed into it. And again, remember, our bile goes in this direction and blood goes in this direction. Notice they've given us a nice job of showing us our sinusoid capillaries. So we have those sinusoid capillaries in between as well. I think if you look at the key for this, notice how there are some cells drawn on this one. I think they've at one point tried to say that those are like the Kupffer cells, but really, if you think about it, the Kupffer cells would be on the inside. So this model doesn't really show the Kupffer cells. but we know they would be here inside of the sinusoid capillaries. But so this model really does an excellent job of doing the histology for this in a nice, clear, and structured manner. And then we also did the histology as well, and we're able to recognize all of these structures, with the exception of the Bile Canaliculi. We were able to recognize all of these structures on the histology slide as well. All right, so I have provided you with these images now for you to study. We have talked about them. They are fair game on the exam, and the fact that I've taken time to do this should be a dead giveaway to you that this is absolutely going to be on the exam. All right, or at least let me rephrase that, because again, the way this exam is set up, everybody's exam is random, so there are absolutely pictures of those models in there. Whether or not you get one picture of them or 5 pictures of them or no pictures of them is completely random. So again, this whole thing is really screwy, but it is definitely something you could be responsible for, and there are a fair number of images on them in the bank. I was working on this, and I still haven't quite finished it yet, but I have over 200 lab exam questions already made and put into the test bank for your lab exam, of which you're getting 65. So everybody's going to have a diversely different lab exam, and it should be interesting to see how that works out. And by interesting, I mean, you know, horribly stupid that you guys are doing this, but that is the format. All right, excellent. We did that, we did that, we did that, we did all of 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 that. Excellent. As I mentioned, and here we see a nicer illustration that kind of emphasizes just the duct work of this. Remember, we have this big, huge triangular liver sitting here. Oops, actually this way, a big right here. And as we talked about, the right and the left hepatic ducts receive from the right and the left lobes. They feed into the common hepatic ducts. It feeds into one of two things, either the cystic duct, which can go up to the gallbladder, or can come out. So again, remember that cystic duct is the only one where the bile can flow in both directions. And the gallbladder and the liver combine to produce the bile duct. The bile duct then expresses out that same hepatopancreatic sphincter that it shares with the primary pancreatic duct. All right, and remember, the accessory is separate. So again, we saw all of that, nothing new there. All righty, clear drawings there, bang, bang. And we already looked at the anatomy of that and talked about that. All right, like I said, we literally could spend the rest of the class time, the rest of this week, talking about the liver. We're not going to do that, so I'm going to talk in very broad strokes. The liver is incredibly active, incredibly busy. Not surprising, it's the largest visceral organ in an adult. It has over 200 functions, but in simplest terms, those 200 functions basically fall under 3 somewhat related categories. The first is metabolic regulation. The second is hematological regulation, and the third is bile production. Bile production is its own thing. Metabolic regulation and hematological regulation are similar to each other. Hematological regulation is more precisely having to do with the condition of the blood, whereas metabolic is more about making the blood appropriate to help other organs, right? So making sure it has the right nutrients and things along these lines. Obviously, what's good for the blood is good for the body. So not surprisingly, these are a little bit overlapping in their function. And I'm just going to rattle them off real quickly. Your book does a decent job of talking about them, so you should have some familiarity with this. But like I said, we're not going to go into massive detail about these processes, right? From a metabolic standpoint, the liver regulates the composition of the blood, as we talked about. One of its major functions is to extract nutrients and or toxins from our digestive tract. So again, all of the blood from our digestive system goes to the liver first, allowing it to make it nutrient appropriate, right? You hopefully had a nice big cheeseburger for breakfast, so there's a large amount of nutrients in your digestive system, and it is flowing through your blood straight to your liver, and your liver storing mass amounts of it. by the time I'm done talking 4 hours from now, right, hopefully your stomach is going to be empty at that point. And so again, the blood isn't going to be as nutrient rich coming to your liver. And so your liver has the ability to provide nutrients to replace those that are deficient. And again, making it appropriate means maintaining blood glucose levels, but also lipids and amino acid levels. And as we talked about, it plays an important role in helping to neutralize metabolic wastes, nitrogen-based wastes, like things like lactic acid or biphosphoglycerate, which we talked about, which is a metabolic waste. Remember, we talked about with red blood cells. Toxins that come in from the foods that you ingest, drugs, medications, recreational, all of those types of things, as well as, again, it is going to play an important role in both breaking down hormones that are produced from other systems, but it also helps to regulate others. Remember, we talked about, for instance, the liver produces angiotensin 1, which, remember, gets activated into angiotensin 2 in the lungs. So again, there is both producing hormones and also breaking down hormones from other systems as well. Metallurgical regulation, again, as we talked about, our blood is a reservoir. So it's circulating throughout the entire body. So it plays a huge role in our immune response. And having those Kupffer cells allow them to break down harmful pathogens, but also, even more importantly, express those antigens on their surface to activate our helper T cells. Remember, that's what is activated by an antigen-presenting cell to stimulate an immune response. It also helps to play an important role in maintaining the osmolarity of the blood. We've talked about the importance of that. It produces an important plasma protein called albumin, which is one of the more common proteins in our blood. And like I said, again, it is by maintaining the blood, it is going to do things like breaking down hormones and toxins and drugs, breaking down antibodies, right, and things like that. And like I said, how you are administered a drug is primarily dependent, and your dosage is primarily dependent on the efficiency of your liver breaking it down. If you look at, and again, one of these classic, you see it more in veterinarianism than you do in humans. But when it comes to giving a medication to a mouse and to an elephant, obviously you give more medicine to an elephant than you do to a mouse, right? The size pills you would have them take is dramatically different. But when it comes to the relation to the body weight or the body mass, you actually give the mouse a much larger dose. A mouse gets a much larger dose of medicine than an elephant does. And you see this with kids as well. Typically, kids get a larger dose of medicine quantity-wise than you do with adults. And the reason for that is that mouse is smaller. The blood passes through the body more rapidly and through the liver more rapidly. They typically have a higher metabolism. So a mouse breaks down their medication much more quickly than an elephant does. So relative to their size, the mouse gets a larger dose of medicine than an elephant does. All right. And like I said, if doing all that blood work wasn't enough, then our liver is also responsible for producing our bile. All right. Again, depending on the composition of your food, the volume of the food, the number of times you eat, it can vary. But on average, our liver produces about 1 liter of bile during the course of the day. Doesn't necessarily mean it all gets expressed during the course of the day, because remember, some of that will be sent to the gallbladder, where the gallbladder stores and also concentrates it. So draws the water out. Again, as I said, it can actually be as much as 10 times more concentrated in the gallbladder than it was when it was first produced. And again, bile production is controlled by lipid ingestion. Remember, the presence of lipids, the presence of those fatty acids causes our enteroendocrine cells in our small intestine to produce cholecystokinin. And that cholecystokinin tells the liver to produce bile, tells the gallbladder to release it, opens that hepatopancreatic sphincter to release those out. Now, what is bile? Bile is a combination of water, cholesterol, and pigments Bilirubin, remember, as we talked about, is a pigment that comes from the breakdown of red blood cells. It's what gives it its kind of yellowish, almost greenish characteristic to the bile. But the other thing that has it that's really important are these bile salts. These bile salts, as we talked about, are what are going to be important for the emulsification of our fat. And they also play an important role in the absorption of our fats as well, as we'll see. Bilirubin, as we talked about, gets broken down into stercobilin and urobilin. Stercobilin is a brown pigment. I think we've talked about this all before. We did in the urinary system. Is a brown pigment. It stays in the digestive system and plays a role in the coloration of our feces, whereas urobilin is actually a yellow pigment. And if you remember, that yellow pigment is actually reabsorbed in the digestive system, so it does come back in the digestive system and is actually released in the urinary system. So the yellow coloration of our urine, as well as the brown coloration of our feces, both come from bilirubin, from the breakdown of those red blood cells. All right. And then, as I mentioned, we'll talk about this process. Our bile, as I talked about, those bile salts in particular, emulsifies fats. Basically, it takes, like I said, one big glob of proteins, of lipids, and breaks it down into small pieces. And the reason for that, if you think about it, is basically surface area. Right? If you've got those enzymes, and remember those enzymes are like those little Pac-Men trying to break bonds, if that fat is all globbed together, and remember also, these fats are hydrophobic. They don't like water. So they all kind of huddle together in mass, forming this big, huge glob of fat. And that makes it very hard for the enzymes to get in there and break the bonds. But if we can emulsify the fat into a whole bunch of little tiny little things, these have a much larger surface to volume ratio, and it makes it much easier for that Pac-Man to be able to break those bonds. And again, hopefully you learned this while you were playing with your Physio-X. Oops, I guess I could sneak and put that up there. There we go. What's interesting about the bile salts, as I said, is not only do they emulsify the fats, but they are also going to play an important role in the absorption of those fats as well. And so what that means is that bile salt gets released by the liver, by the gallbladder into the small intestine, where it breaks down those lipids, gets reabsorbed into the blood supply. And when it gets reabsorbed into the blood supply, what's the first place where that blood goes?
To the liver.
To the liver, absolutely. And once it gets to the liver can actually collect those bile salts and actually release them again. Again, depending on the size of the meal, the composition of your meal, a single bile salt can actually be recycled and reused as many as 5 times in the digestion of a meal. So again, it can be used many, many times during this. And what's cool about it is it's a bit of a positive reflex process. I've got a pretty picture that shows this in a minute, but I'll say it here out loud. So there is fat in your small intestine. Actually, let's look at the picture. We'll go through it that way. That'll make more sense. So hold on, let's go ahead and move. There we go. That's a pretty picture I want. All right. So let's think about what's happening here. All right. It all starts, and I'll just do numbers. Number 1. Here. Oh, actually, they are numbered. There. That makes sense. Here, our chyme is presented into the small intestine. When the chyme is presented in the small intestine, remember that chyme has fatty acids in it, and those fatty acids stimulate those enteroendocrine cells to produce the hormone cholecystokinin. that cholecystokinin gets into the blood supply, and conveniently enough, the blood supply of the small intestine goes first to the liver. So we have this blood supply going to the liver, and when it goes to the liver, this cholecystokinin tells the liver to produce bile, tells the gallbladder to contract and release bile. I guess the liver is producing and releasing. And of course, it tells the valve here, the hepatopancreatic sphincter valve, to relax so that we can release that bile into the small intestine. That bile is released into the small intestine, mixes with the fats, and then that bile gets reabsorbed. And when it gets reabsorbed, what happens is that bile goes back to the liver. And when it goes back to the liver, that bile going back to the liver, when the liver notices that there is bile in the blood, that bile salt in the blood stimulates the liver to make more. And when it makes more bile, it releases more bile. And so more bile goes into the small intestine, mixing with the fats, gets absorbed, and that bile coming back stimulates more and more and more and more. And so in this fashion, as long as there is fat in the small intestine, our liver can produce and release more and more bile. Now notice, when there's no more fat left, there's nothing for that bile to attach to, and the bile can't be reabsorbed. And if it can't be reabsorbed, less bile goes back to the liver, and as less bile goes back to the liver, it produces and releases less. As there's no lipids left in here, our enteroendocrine cells stop producing cholecystokinin, no more stimulation to the liver. And so once those lipids go away, bile production and bile release decreases and stops as well. So it's this great system where it can ramp up as long as there's lipids there, and then it'll ramp back down as soon as those lipids go away. All righty, and the cool thing about that is, like I said, those bile salts can be reused again and again and again, over 5 times during a single meal, depending on the composition of it. All righty, questions on that? All right, excellent. So, cool. We're at a good stopping point because when we come back, we have our next major process. As I told you about in the last class, these absorptive pathways are huge and important, and I guarantee one of these 4 pathways will be an essay question on your exam. So all 4 of them are in a bank. One of them will be randomly selected. So I guarantee will have one of these questions on the exam. Our goal in the small intestine, as we said, the small intestine is the major site of absorption. Notice we've talked about other places. Absorption occurs in the mouth. Absorption occurs in the stomach. There is some absorption that occurs in the large intestine as well, but the small intestine is the major site. Our goal has been to take those large, complex molecules and break them down, again, chemically by enzymes. Know your enzymes that break them down. You need to know those. And of course, there are those enterocytes, those brush border enzymes that are located on our small intestine as well. You don't need to know the names of the individual ones for those, but anything else we've talked about, trypsin, pepsin, lingual lipase, all of those you definitely need to know. And large molecules are broken down into their small building blocks. As we've talked about, our proteins are broken down into amino acids. Our polysaccharides are broken down into simple sugars and monosaccharides. Our lipids are broken down into alcohols and fatty acids. And of course, I still spelled lipids wrong. There we go. And of course, our nucleic acids are broken down into nucleotides. So again, all of our large macromolecules are broken down into those basic building blocks, and it's these basic building blocks that are going to be absorbed. And if you look here, we have there you go, and like I said, 90% of the materials are absorbed in the small intestine, most of it in the proximal end. And if we look here, we see the simple version of this process and a framework of what we are going to be responsible for. All right, we have these large macromolecules that are broken down into their basic bits. They then need to, just like we saw in the urinary system, pass through two membranes, the apical membrane of an absorptive cell and the basal membrane of the absorptive cell. And often the mechanism on both surfaces are going to be different. Once it is in the interstitial fluid of our small intestine, then it is either going to enter into our blood supply or a lymphatic supply. And because it's information we already need to know, once it's into either the lacteal or into the blood capillary, we then need to trace the path to the right atrium, because we know both of those paths and we will be doing that as well. And so after we take a nice break to recharge, get a cup of coffee so that you're amped up and ready to go, right? We are going to take a quick 10-minute break and come back, and then we will do these pathways. All right. Questions on anything we talked about the liver before we take our break and then move on to this fun stuff? All right. My watch says 9:22. So let us restart at 9:32, and then I will start the recording again at that point. All right, I'm going to go ahead and pause the recording now. So let's dive in. Our goal, oops, clear the picture, is to, again, talk about the absorption. We need to talk about all of the stuff that's going to go on to break it down to its basic building blocks. But ultimately, as we've talked about before, and again, this is somewhat similar to what we talked about in our urinary system. We have these absorptive cells, and these absorptive cells have microvilli on the apical surface. And in kind of a similar fashion to what we talked about, what is out here in the lumen, in this case, the lumen of our digestive system, is literally outside the body. So what that means is if we want to absorb something, we have to first get it through that apical surface. Get it into the cell. Once it's into the cell, we've absorbed it into the body. At this point, technically, it's no longer outside the body, it's in. But if we're going to use this stuff, we then have to get it out of the cell, out of the basal surface. And once it's out the basal surface, then like I said, first we are getting it into the interstitial fluid. But once it's here in the interstitial fluid, ultimately our goal is to distribute it throughout the body. So once we get it from the interstitial fluid, eventually we want to get it to the heart. And so, because once it gets to the heart, it can be sent anywhere. And so that's going to be our goal, right? Is to get it across those two surfaces. So just like we saw, and again, normally remember this comes before urinary system. So we've had a chance to talk about some of these things for the urinary system first. So again, when it comes to membrane transports, we have active and we have passive. Especially on the apical surface, we are going to want to use active processes. And those active processes, of course, use ATP, either directly or indirectly. And notice we're talking about co-transporters. So, of course, those indirectly use ATP. Whereas once it's inside, we can use things like facilitated diffusion to be able to get it as well. But both of these will be used in these fashion. And again, typically what we use on the apical surface is not what we're going to do on the basal surface. So again, go back to chapter 3. Hopefully you did all of that for the urinary system. So, and again, like we said, once it's into the interstitial fluid, where does it go and how do we get it to our heart? That's going to be the goal. All right. So I've got the pretty picture here, but I think what I want to do is switch to the whiteboard for this. Because ideally, the way you're going to set this up is going to be similar for all of these. All right, so if we think about it, and let's go with something nice and big and black. And I think we're going to need some room to write, so I think that'll be the good font. All right, so first, obviously, we have our macromolecule we are going to be interested in. From there, we also need to know what it's broken down into. All right. And when we talk about breaking these things down, remember we're going to talk about both chemical digestion. Oh, let's do it this way. Mechanical digestion. And chemical digestion. All right. And after that chemical and mechanical digestion, we want to identify all the locations where these occur and how it is done. Once we do that, we need to talk about how it passes the apical surface. Once we do that, we want to say what occurs inside the cell. Which sounds weird, but we'll see, isn't really. And then how it passes the basal surface. Then once it passes the basal surface, we want to know the type of vessel. It is collected by. And then once we're done that, we want to, and let's do it here, pathway to the heart, or more specifically to the right atrium. Oops. Excellent. That's all you need to know. Looks super simple, doesn't it? And it is totally superly simple. So, like I said, we are going to start first with, hold on, oops. These pathways. So again, when we're talking about these pathways, we need to be able to see how this is going to go. Wait, what am I doing? What can you guys see?
The synapse.
Oh, this isn't sharing at all. Okay, I'm sorry. Hold on. Okay, sorry. All right, let's try this again. Can you see that now, the whiteboard back?
Yes.
Yes.
Okay. All right. So let's go through this. And this, again, this is our building, this is our framework for everything that we're going to do. And remember, as we talked about, we're going to start with the carbohydrates. Right, that is our macromolecule. And of course, carbohydrates are broken down into what?
Monosaccharides.
Yeah, monosaccharides, right? Simple sugars. which are really our disaccharides, but also our monosaccharides. Excellent. So that is our first goal. Let's talk about where mechanical digestion occurs. Where is the first place that our carbohydrates are broken down mechanically?
In the mouth.
Excellent. And what is the process that occurs in the mouth that does that?
Mastication.
Yeah, exactly. Chewing, mastication. There you go. Absolutely. Right? All of you, as we talked about, all chew our foods the 35 times you're supposed to chew them before you swallow it down. However, if you don't, right, luckily mechanical digestion occurs other places, like where?
Stomach.
Stomach.
Yeah, what happens? What do we call that process in the stomach?
Churning.
Yeah, there you go. Churning that occurs in the stomach. Is there any other type of mechanical digestion that is taking place?
Hydrochloric acid.
Well, remember, hydrochloric acid, you are absolutely correct, is a form of mechanical digestion where it denatures. However, what is it that hydrochloric acid denatures?
Proteins.
Protein. So actually that hydrochloric acid doesn't have a significant mechanical effect on our carbohydrates. So you're absolutely right. We're going to use hydrochloric acid as a mechanical digestion, but not for carbohydrates. That's going to be for proteins. But the other one is in our small intestine. Remember, we have that segmentation. Segmentation, remember, is that alternating contractions of the small intestine, not so much for propulsion, but squishing the food back and forth. As we talked about, that is going to mix it with the enzymes to facilitate chemical digestion. It is going to rub it on the surface for absorption, but the other thing it does is mechanically break it down. So those are our big three for mechanical digestion. And notice we could have done that in permanent marker up here, because if you think about it, for all of our macromolecules, there's mastication, churning, and segmentation. So those are really, those three are the same for everything. All right. What about chemical digestion? Where does chemical digestion begin for our carbohydrates again?
In the mouth.
The mouth.
Mouth.
And what is the enzyme that does it?
Salivary amylase.
Salivary amylase.
Excellent. All right. Let's work our way through the pathway. Any chemical digestion occurring in the stomach for carbohydrates?
No.
No. So that brings us to the stomach. And pardon me, not the stomach. I just passed the stomach. The small intestine. And in the small intestine, what are the enzymes going to be that are going to facilitate the chemical breakdown of carbohydrates?
Pancreatic amylase.
Excellent. And remember, we also talked about, even though you don't need to know the individual names, we want to put those brush border enzymes in here as well. So after chewing and churning and segmentation, after salivary amylase, pancreatic amylase, and brush border enzymes, all of our carbohydrates now in our small intestine are broken down into simple sugars. Monosaccharides and a few disaccharides can be absorbed as well, but primarily monosaccharides. Now, not surprisingly, especially now that we are all home and quarantined, we have a carbohydrate-rich diet. So getting carbohydrates isn't a major concern. So when it comes to crossing the apical surface, we, of course, in a similar fashion to what we saw in the urinary system, are going to use secondary active transporters, primarily co-transporters, to bring those carbohydrates, those simple sugars, those monosaccharides now across the apical surface. But like I said, our diet is so carbohydrate rich and it's not so much an issue. We can also use facilitated diffusion as well. So there's just some simple facilitated diffusion carriers can be used to bring it in through the apical surface. Once it's inside the cell, again, it is just going to sit there and not do anything. So really, there is what happens inside the cell is not applicable here for our carbohydrates. But they are going to accumulate in this absorptive cell and eventually want to leave. So on the basal surface, but like we've talked about before, we wanted to use energy to be able to capture it from our digestive system, bring it into the body. But are we going to want to necessarily worry about using energy down here on the basal surface? No. So instead here, we are just going to go ahead and use facilitated diffusion. And with that facilitated diffusion, it then ends into the interstitial fluid. Now, once it's in the interstitial fluid, what is the vessel that collects it, right? There's basically two choices. If you think of that villi, right, that villi has a blood vessel capillary in it, but it also has a lacteal. Which of those two structures are our sugars going to go into?
Blood vessel.
There you go, into the blood, let's say it this way, blood capillary. Excellent, and once it gets, 1P, 2Ls. Once it gets into that capillary, right, what is the path that this is going to take to get to the right atrium of the heart? That blood capillary is going to feed into what blood vessel?
Hepatic portal vein.
It will, but what does it feed into before? So let's put that. However, what feeds into the hepatic portal vein?
The mesenteric veins.
True, although more specifically, the intestinal. Right? We're in the small intestine after all. So that intestinal vein is going to feed into the hepatic portal vein, which feeds into what? We just learned the macro anatomy. Our hepatic portal veins feed into what?
The liver.
Hepatic vein.
Not before it gets to hepatic vein, though. Right before we just said magic happened, but now we actually know. Again, this is a portal system that usually involves two capillaries. The first capillary was before the intestinal vein. This is going to then feed into that sinusoid capillary of a liver lobule, and that sinusoid capillary feeds into what?
Central vein.
There you go. That central vein, which feeds into the
Hepatic vein.
go, which feeds into the
Inferior vena cava.
which feeds into the
Right atrium.
there you go. And lo and behold, we made it. So just that simply, we have described everything that happens to that carbohydrate from the second that you stick it in your mouth and ingest it to the point where it gets as a simple sugar, as a glucose into your right atrium, where it can be distributed throughout the body. That makes sense? Stunned silence means yes, so excellent. We will continue forward. We just described it. Let's do, hold on, I have a chat thing which apparently was down. Bring my chat up. Where was that? Come on, there it is. Can I screenshot that? Sure, I can do that. All righty. Although you should be writing this down anyway. So, like I said, here we can do the exact same thing with our carbohydrates broken down into our simple sugars. Again, those monosaccharides. Why is that not working? I'm so confused. Oh, I'm not sharing this. That's why. All right, excellent. Now it'll work. Excellent. So broken down into simple sugars, and we've identified the locations. Notice your book has a nice, pretty flow chart that kind of shows this. So again, our carbohydrates in our oral cavity broken down by our salivary amylase into our stomach. Right now, we now have some triglycerides and disaccharides into our small intestine, where our pancreatic amylase is yeah, because it's not applicable. Because again, it's a weird thing to say that something might happen to it inside the absorptive cell. But as it turns out, for one of our macromolecules, something interesting does happen inside of the cell. So I wanted that as a placeholder because one of our 3 pathways we're about to talk about right now, something does happen. So we, again, pancreatic amylase, our brush border enzymes, break them down to those simple sugars. Our lactase, our maltase, our sucrase, those are some examples of brush border enzymes. They break them down into simple sugars. So again, chewing, churning, segmentation, enzyme breaks them down into those monosaccharides. Once we have those monosaccharides, they cross the apical surface by facilitated diffusion and a co-transportation. Once inside the cell, they accumulate, they leave via facilitated diffusion, enter into a blood vessel capillary, and we have followed the path to our heart. All right. So again, no new information here, just another pretty way of showing this. And again, your book's got a couple tables, a couple illustrations that do a decent job of showing us what that is. Why do my chat window keep going away? That's weird. Can I get that to come up? There it goes. Okay, perfect. All righty, excellent. So now that we've done that, we can do the same thing with our proteins as well. All right. So, oh, and here again, have the pretty pictures, but let's stop it. There we go. Okay. So let's go back to the whiteboard. You guys can see the whiteboard now, right?
Yeah.
Yes.
Perfect. So obviously that part goes away. That part goes away. That part, that part, that part, that part, that part. Although really those last two don't, but that's okay. We'll do it again anyway. Let's do the same thing now for proteins. Proteins, of course, get broken down into what?
Amino acids.
Into amino acids. Notice mastication, churning, segmentation, as we mentioned, are going to be the same for all of our macromolecules. However, as someone mentioned before, we get to add a fourth. And what was that fourth again?
Hydrochloric acid.
Yeah, the hydrochloric acid. Absolutely. That hydrochloric acid, as we talked about, is a denaturing the proteins. And again, remember that denaturing is an unfolding of it. It doesn't cut the bonds, but it does unfold it to make it easier to cut the bonds. So since it is changing the shape without breaking bonds, it is considered mechanical digestion. Where does our chemical digestion begin?
In the stomach.
Excellent. And what was the enzyme?
Pancreatic.
Close. Pepsin. Pepsin, absolutely. In our stomach with pepsin, right? Again, that whole hydrochloric acid denatures, so then those pepsin Pac-Men can get in there and break the bonds. But also in our small intestine, we are going to use enzymes there as well. And of course, what are the enzymes there? What's the primary one?
Trypsin.
Trypsin and chymotrypsin and carboxypeptidase, those as well. I'm not going to bother writing those out. And also, again, our brush border enzymes. So again, Using this mechanical digestion, using this chemical digestion, we break things down into amino acids. Now, we got to get those amino acids into our body, out of the lumen of our digestive system. And if you remember way back in 430, when we first started talking about cells, we said cells are protein factories. When we talked about proteins, when we talked about our macromolecules in 430, remember what we talked about is that 20% of your body weight is proteins, which I know on the surface doesn't sound like a lot until you remember that 60% of your body weight is water. So you're only 40% stuff, and half that stuff is proteins. Proteins are hugely, massively important. So it is hugely, massively important that we get them into our body. So we're not going to just rely on luck or rely on hope. We are going to use energy. So we are going to use both primary and secondary active transporters. We will use co-transporters like with sodium and other things to get it in, but we're also going to have pumps that are just going to pump those amino acids out of the lumen and into our body. Again, we don't have to do anything, so not applicable. Remember, that's what Na stands for. There isn't anything fancy we need to do inside the cell with those amino acids once they're inside, but once they're inside, they've been saved. However, we still need to get them out. It is important to get them out and into the blood. So on the basal surface, we will use facilitated diffusion, all right, some carriers and things like that, but we also will use some co-transporters as well, because we want to make sure we get that amino acid out into the interstitial fluid. And of course, once we get it out into the interstitial fluid, which of our two vessels does it end up in?
Capillary.
Yep, blood vessel capillary. Remember, technically a lacteal is a capillary as well. So you don't want to just say capillary, because there are lymphatic capillaries and blood vessel capillaries. So we want a blood capillary. And that blood capillary, and this should hopefully be easier this time, the blood capillary feeds into
Intestinal vein.
intestinal vein, which feeds into
Hepatic portal vein.
hepatic portal vein, which I'm going to abbreviate because we already did this once, which feeds into, actually I'm not, because I want to make sure to emphasize, hepatic portal vein, which feeds into,
Sinusoid capillary.
yeah, sinusoid capillary, which feeds into
Central vein.
central vein, which feeds into
The hepatic vein.
hepatic vein, which feeds into
Inferior vena cava.
and then that feeds into the right atrium. So I'll abbreviate it there. Excellent. Again, that should have been easier because it was the exact same pathway that our carbohydrate took. All right. So there you go. With a simple chart, we once again, and someone asked for a screenshot, so I assume you'll want one for this as well. So there you go. We did that. And if we go back to here, again, broken down into amino acids, right? We talked about in the stomach. We talked about in the small intestine how that occurs. And again, here in our stomach, pepsin breaks it down, churning. Of course, we have mastication that occurs up in the oral cavity. In the small intestine, we have trypsin, carboxypeptidase, chymotrypsin, all of those. Elastase, which is another one we didn't mention, breaks it down into those small short peptides and amino acids. And then again, we have those brush border enzymes that are going to break it down into amino acids. A dipeptidase is a cool one. Basically, all a dipeptidase does, and again, you don't have to know this name, but what's cool about it is basically it just finds an amino acid and whatever it's bound to, it breaks it. So it finds a sequence of amino acids and doesn't care what the amino acids is. It just finds the end and then just starts snipping them off. So we get those simple amino acids, and we want those amino acids. We need those amino acids. So there's no ifs, ands, or but about it. We are going to use co-transporters. We are going to use active transporters. I know your book says facilitated diffusion, but one of the things that I really want to emphasize about this is that this is important. So we are going to use energy. Yes, things find a way to sneak their way in. These pathways are not completely impermeable. So yes, some diffusion could come in, but that's not the main way we're getting it in. Our goal here is to use energy and make sure we get those amino acids across. And not only do we use energy to get it inside, but we use energy on the other side to get it out as well. We're going to use facilitated diffusion and co-transport on the basal surface as well. And as we talked about, once it's out, it is into that capillary, and we know the pathway of the blood system back to the heart from there. Your book, again, has a nice, pretty chart that talks about these things and where the enzymes are and what's going on and how that's occurring. It also, and I don't know why it doesn't do this for the carbohydrates, it also shows a pretty picture showing this process of the passageway across it as well. So here are our brush border enzymes. Here are pancreatic enzymes breaking those polypeptides into amino acids. Here we have a sodium co-transporter bringing it in this way, and then we have a facilitated diffusion coming out the basal surface and right into the capillary. So we have that nice pretty picture that shows that as well. All right, those are simple, those are straightforward. Now's where things get fun. Lipids. So questions on proteins before we switch to the third. All right, excellent. Let's switch to the third. And so again, get rid of that, get rid of that, get rid of that, that, and that. And now we are ready. Oops, why does that still say proteins? You don't see where it says proteins at the top, do you?
No.
Okay, I can still see a little bit of the slide behind it. So, so I just wanted to make sure that wasn't confusing for you guys as well. All right, we haven't used blue yet. Let's use blue. Give me my text. Give me blue. Our goal here now is to break down our lipids. And as we already talked about, lipids are broken down into alcohols like glycerol and fatty acids. All right. Now, the big three, mechanical digestion is still there. You're still chewing, you're still churning, you're still segmenting. Is there a fourth mechanical digestion that takes place?
Bile salts.
There you go, absolutely. Our bile salts. And of course, where does that occur?
Small intestine.
There you go, small intestine. Although we should, in parentheses here, probably put liver/gallbladder, because those are the ones that are providing the bile salts, but the actual act of breaking it down is occurring in the small intestine. And there in the small intestine, our bile salts emulsifies the fat. So we have that emulsification, all right, from the bile salt. So that is our fourth mechanical digestion that takes place. From a chemical digestion standpoint, where does chemical digestion of fats begin?
In the mouth.
Oh, careful. Again, where does it actually physically begin?
Small intestine.
Nope, in between. You guys have the right idea. Stomach, but way of the tongue. And that's why, again, I know it's a little tricky. So if you think about it, remember the tongue is producing that lingual lipase. It is producing the lingual lipase, but remember, it's not active in the mouth. It doesn't become active till it reaches the stomach. So the same way that, for instance, it's our pancreas that is providing the enzymes to break things down in the small intestine, it is the tongue, the mouth, the saliva that is producing that is providing the enzyme, but it doesn't actually become active till the stomach. So it starts in the stomach, continues in the small intestine, And as you guys already mentioned, that is going to be pancreatic lipase. So we have those lipases, that are going to break those. And again, also, that's not an and, those brush border enzymes that will help. All right. Now, here's where things get a little bit interesting. As we talked about, our bile salts are going to emulsify that fat. But not only do they emulsify the fat, but when the lipases break it down into the fatty acids and the alcohols, the bile salts are going to bind to the fatty acids, right? And when they do that, they form a structure called a micelle. So these micelles are a special structure. Now, they're fatty acids, right? We also have, like I said, alcohols. But these micelles and these alcohols are both lipid-based. And if they're lipid-based, what does a lipid-based substance need to do to get past that lipid-based apical surface?
To put proteins around it.
Yeah, it really doesn't have to do much of anything because it's lipids, lipids like lipids. It is basically going to be able to pass via just simple diffusion. We don't need any proteins. We don't need any special transporters or anything like that. Lipids like lipids, and so it is going to pass through just via simple diffusion. So the alcohols and those micelles are going to enter into the cell via simple diffusion. Now, once inside, the bile salts and the fatty acids, I'm just going to abbreviate that FAs, separate. And I'm going to come up here and I'm going to emphasize this by changing to a different color. Hey, green's convenient. Bile salts, remember, are going to be via facilitated diffusion. Leave the cell, leave the basal surface, enter into the blood capillary, and are transported back to the liver, right? Where they can be recycled and used again. So again, remember, as we already talked about how the bile salts are going to leave, be reabsorbed, and be used by the liver a second time. All right. However, what happens here, and I need to go back to blue. So that's what happens to the bile salts. But what happens here is the fatty acids and the alcohols reform into triglycerides and other lipids. Notice when we talked about our carbohydrates, when we talked about our proteins, once they got inside the cell, they just diffused out the other end. That is not what happens with our lipids. We emulsify them, we break them down into their basic building blocks so we can passively absorb them on the apical surface. But we don't want to take those lipids and release them into the interstitial fluid. We don't want lipids running freely inside the fluids of our body because lipids and water don't like each other. And so we don't want just random fatty acids and random other alcohols running around all over the place inside of our blood and other structures. So instead, we need to organize them. And so that's where this is special. Inside that absorptive cell, the fatty acids and the alcohols come back together and form our lipids. Once they do that, they bind with other lipids and form these big structures. But again, a big massive lipid isn't necessarily a good thing to have inside the blood either. So we need to make it water soluble. And so the way we do that is we coat the outer surface with proteins. And when we coat the outer surface with proteins, now it is water soluble. And this water soluble structure is called a chylomicron. And it is this chylomicron, and actually I need to give us a little bit more space. So I'm going to sneak this up a little bit there, sneak that up a little bit there. So we have now made this chylomicron, this big, huge, massive clump of fats with a protein coat on the outer surface to make it water soluble. Now, once that chylomicron is made, do you think we can have a big, huge hole in the basal surface of the cell to let this thing out?
No.
No, that would affect the permeability of the cell. So to get it out on the basal surface, we actually need to use a mechanism we use for releasing large molecules or large amounts, right? Vesicle transport. So the short version of how it passes the basal surface is exocytosis. Oops, that didn't need to be capitalized. So it is released, let's say it that way, via exocytosis into the interstitial fluid. Now, even though it's got this protein coat of water-soluble proteins on the outer surface, and so it is water-soluble, do we necessarily want this big, huge mass dumped into the blood vessel supply?
No.
No. So where does this chylomicron go? What vessel is it collected by?
The lacteal.
Into the lacteal, absolutely. Luckily for us, we talked about the lacteal circulation, the lymphatic circulation, so we can follow that pathway in that lacteal, from the lacteal into its collecting vessels. And of course, those collecting vessels are both efferent and efferent and feed into the lymph nodes. And those collecting vessels from the digestive system is going to feed into one of our 9 trunks. Which of the 9 trunks is these collecting vessels from the small intestine going to feed into? If only I remembered my lymphatic trunks. Give you a hint, it's not the subclavian or the jugular or the bronchomediastinal. Say again?
Thoracic.
We're going to get to the thoracic duct. Remember, there are 2 ducts. So we are going to get to the thoracic duct. You are right. That is going to be the end of our story, or near the end of our story, is to get to the duct. But we need a trunk to get there first. Does anybody remember the trunk of lymphatic trunk that would come out of, oh, say, for instance, the small intestine or the large intestine?
Intestinal duct.
There you go, trunk. Exactly. Intestinal trunk. But remember also, the intestinal trunk doesn't feed directly into the thoracic duct. There was a structure in between. What was that structure in between?
Cisterna chili.
Say again?
The cisterna chili.
There you go. Cisterna chili. Right? And that cisterna chili feeds into the thoracic duct. The thoracic duct accumulates lymph from the left side of the body and the abdominal region and the legs, and it feeds that lymph into what structure? Anybody remember what the thoracic duct feeds? Because remember, the goal of this lymph is to eventually get it back into the blood supply. Where does the thoracic duct dump its lymph into the blood supply?
Subclavian.
There you go. More specifically, the left subclavian vein, which feeds into what blood vessel?
Superior vena cava.
Before that? The left subclavian and the left internal jugular come together to form a blood vessel that collects blood from both the head and the arm. So that would be the left. No one. Trying to make it easy. Left brachiocephalic vein. And then you're right. The left brachiocephalic vein feeds into the superior vena cava, which of course then feeds into the right atrium. And lo and behold, using knowledge we've gained along the path in this classroom, we are now able to follow the path that that lipid takes to get back to the heart. All right, questions on that one? All right, we've done it here with the pretty words. Let's take a look at the pretty pictures. Again, lipids are broken down into alcohols and fatty acids. Again, that lingual lipase is released in the oral cavity, but remember, doesn't become active till it gets to the stomach. Our bile salts and our pancreatic lipase, right? Our most common dietary lipid are triglycerides. So those are broken down into fatty acids, into alcohols, like monoglycerides and stuff like that. They then mix with those bile salts to form our micelles. And those micelles, as we talked about, are going to pass the apical surface just via simple diffusion. So it is those monoglycerides, those fatty acids that enter into our cell. And once in the cell, we reform them into our lipids. So our triglycerides, our steroids, all the things like that, all of these lipids are formed. They are then bound with other lipids like phospholipids and steroids and other things, and coated with those proteins to form that chylomicron. That chylomicron is a huge molecule, so there's no way to have a channel to let it out, so we have to release it via exocytosis into that lacteal, which flows back through our lymphatic vessels into our cardiovascular system and back to the heart. Again, we've done it with words. We've done it with this nice chart. And again, your book's got the nice chart. Your book has this great picture, which I really like. It really does a good job of showing this with one major, and in my eye, really confusing decision that they've made. Notice they do a really great job of showing the emulsification of our fat. We have this big globule of fat. The bile salts emulsify it. And when they emulsify it into the small bits, so here we have those emulsified fatty acids, it makes it easier for the enzymes to break this down. Once these are broken down into alcohols and fatty acids, they then mix with the bile salts to form our micelles. Our micelles then enter into the cell via simple diffusion and are made up, rebound into, again, the bile salts are released into the blood, back to the liver, but the fatty acids and alcohols are converted back into triglycerides, bound into a into a chylomicron coated with proteins, which releases via exocytosis into the lacteal and on its merry way. So visually, they do a really nice job of showing this. My only major problem with this is if you think about it, in real life, what's bigger, a micelle or a chylomicron?
Chylomicron.
Yeah, chylomicrons are massive. Now, again, I can see what they were trying to do. They were trying to show us all of this in the pathway here. But you got to remember, these things are tiny enough to pass through simple diffusion. These things are so massive, they need exocytosis to get out. So aside from this changing scales on us, they wanted to show the emulsification. They wanted to show the formation of the micelles, which is great. And they also wanted to show the path through the absorptive cell. But by combining these two images into one, they've been a little confusing with the size of this. The only important thing to remember is these things are tiny, these things are huge. But other than that, it really is a nice depiction of this. All right. Questions on that? There you go. Excellent. So those are your pathways, like I said. However, as I mentioned, there is a fourth pathway we need to know. So, of course, what is left?
Nucleic acids or water?
There you go. Nucleic acids is the last macromolecule, but honestly, the path of that is pretty damn boring and almost identical to carbohydrates, and even less so because all the breakdown occurs in the small intestine. So for our fourth pathway, which is much more interesting, is the absorption of water. All right. One of the things to think about is that there is over 9 liters of water that enters into our digestive pathway during the course of the day. Over 9 liters of fluid. However, if you notice, less than 2.5 of that is from the food and from the liquids that we ingest. All along this pathway, whether it was saliva, whether it was gastric juice, or whether it was bile, or pancreatic juice, or intestinal juice, there is, 7 liters worth of fluid that is leaving your body in a 24-hour period of time. Because remember, the lumen of our digestive system is outside of the body. All right. So it's outside the body. So we are producing 7 liters of stuff during the course of the day and releasing it from the body, 7 liters of fluid. Now, how many people have 7 liters of excess fluid in their body that they can afford to lose every day? Not many of us. Luckily, the majority of this is reabsorbed. And as we also talked about, the majority of this water reabsorption occurs in the small intestine. Of the 9+ liters of fluid that enter our gastrointestinal tract during the course of the day, 8 of it or so is absorbed in the small intestine and only 1 in the large intestine. As we talked about in the past, many people incorrectly assume that the large intestine is the primary site of water reabsorption. Water reabsorption does occur in the large intestine and not a whole lot else. So that's why everybody remembers that. Water is reabsorbed in the large intestine and it doesn't do much else. But it's really more just fine tuning. The majority is indeed absorbed in the small intestine. When it comes to pathways for this, it is going to pass through the cell via osmosis on both the apical and basal surface. It is water after all. And again, and in many ways, this is similar. It is not called the same thing, right? Remember, if you back in the urinary system, we called this obligatory water reabsorption. Because all this stuff is being transported in and water has to follow. Essentially, that's what it is. So we'll actually say it. It's similar to our obligatory water reabsorption. It's not called obligatory water reabsorption, but essentially the same thing. Water is following all the stuff that is being absorbed. So it's going to pass both the apical and the basal surface via osmosis. That's pretty simple. Where this pathway is tricky is that, not surprisingly, water enters both the blood vessel capillaries and the lymphatic capillaries. So it is going to take both pathways, both through the liver and through the cisterna chyli, back to the heart. So the absorption is pretty simple and straightforward. Obviously, we're not chewing the fluids, right? In fact, we're secreting most of them. But it's osmosis across both apical and basal surfaces. And then we need to know the 2 pathways. All right, there isn't any breakdown, chemical or mechanical or any of those things. So that part of all of that is easy. The tricky part is that it's both pathways back to the heart. All righty. Questions on that? So our goal is to absorb most of the water, so only about 100 milliliters remains in the feces for expulsion. And with that, we have mostly processed the food, and we are on to our large intestine, and at a good, another natural stopping point. All right, questions on that? So again, one of those four pathways: water, carbohydrates, lipids, or lipids, or proteins. One of those is going to be an essay question. All right, excellent. Let's go ahead and take our next break. Take a 10-minute break. That means coming back at 10:36. And I will start the recording at that time. All right, I'm going to go ahead and pause the recording now. Zoom record. Excellent. All right, so we are most of our way through the alimentary canal, and we have done most of the processing. Like I said, by the time we reach the large intestine, we still have chyme. Remember, we went from food to bolus to chyme. We're still at the chyme, but it's mostly processed chyme. It's basically essentially going to be chyme when it enters the large intestine, but obviously if it leaves the large intestine, it is going to be feces. So there is some last little bit of processing to take place. The large intestine, as we can see, is a relatively unassuming looking organ, but there are a lot of specializations to it, as we can see when we look at the anatomy of this. And where's my annotation tools? Thank you. It starts with a large pouch-like structure. And of course, it starts with a valve that feeds into it. Like most of the organs we've talked about, it is bounded on both ends by a valve, a proximal and a distal valve. In fact, there are two distal valves in an anatomy that we'll see very similar to what we just did a couple weeks ago. We have our ileocecal valve, and it feeds into a pouch-like structure known as the cecum. The cecum then feeds into the ascending colon, into a large bend region, which is known as the right colic flexure, because it is a bend flexure of the colon. However, the right colic flexure, if you think about it, is right next to the liver, and so is also sometimes referred to as the hepatic flexure, and either of those terms are acceptable. It then feeds into the transverse colon, into the left colic flexure. And if you think about it, the left side is where the spleen is located. So this is also known as the splenic flexure. Again, either term is acceptable. That feeds into the descending colon, which feeds into the sigmoid colon, which feeds into the large pouch-like structure known as the rectum. So again, we have a pouch at the beginning, a pouch at the end, a valve at the beginning, two valves at the end. So two pouches, and then the colon in between. So 3 regions, 8 parts to its basic anatomy. As we also talked about, remember both the ascending and the descending colon are retroperitoneal. They're embedded and anchored in place in the abdominal pelvic cavity, whereas the cecum, the transverse colon, and the sigmoid colon are intraperitoneal. All right, I think about the cecum has to be because it connects to the ileum, which is intraperitoneal. And remember also, the transverse colon and the sigmoid colon have their own mesenteries that support them and anchor them in place. They're not shown on this illustration, but remember we have the transverse mesocolon that holds the transverse colon in place, and the sigmoid mesocolon that holds the sigmoid colon in place. The basic anatomy is similar to what we see in the majority of the digestive system, with a mucosa, submucosa, 2 layers to the muscularis, and then either a serosa or an adventitia, depending on if it's intra or intramembranous or intraperitoneal or retroperitoneal. But there are some unique characteristics to this as we look at it. One of these is, if you notice, there is a fair amount of tension in the large intestine. And as you put that tension on it, it causes a pooching or pouching of the large intestine. So if you notice, all of these enlargements, these pouch-like structures, are what known as the haustra. Haustra, of course, being the plural, haustrum being the singular. One of the reasons it pouches is, if you notice, there are these enlarged bands of the longitudinal layer of the muscularis. These pronounced enlarged bands are known as the tinea coli. So the tinea coli basically is an enlarged band of the smooth muscle, specifically of the longitudinal layer of the muscularis. And so these large bands help to condense or shorten the large intestine and form those pouch-like structures. If you remember also, sorry, trying to drink my caffeine, way back in the lymphatic system, we talked about that vermiform appendix, that worm-like structure that connects to the cecum of the large intestine. And we talked about the implications of that for our appendicitis and when that burst and everything that went along with that. But there are a couple other things to notice as well. I don't think it's on the list, but I want to point it out to you right here. Notice cut right here would be that greater omentum. Remember, the greater omentum drapes down and then back up and connects to the greater curvature of the stomach. So we see that as cut. And that mesentery, like many of the mesenteries, will have fat deposits in it. But if you notice also, there are these fat deposits that are actually directly connected to the large intestine itself. These fatty attachments are what are known as the epiploic appendages, or the fatty appendages. Both of those are acceptable. Their functions are not fully understood. They may be storage of nutrients in those areas from excess nutrients. They may play a helpful role in maintaining appropriate fatty acid levels for the bacteria that is in here. Tinea coli and tinea coli is both, one is singular, one's plural. With the E is plural, without the E is singular. So yes, so there technically are two ways, singular and plural, for the spelling of the tinea coli. So that's the difference. And again, remember, you get one letter, so you can technically spell it either way, and it would still be correct. But like I said, the full functions of these epiploic appendages are not fully understood, but they may play a role, like I said, in helping maintaining consistency by storing nutrients or something along those lines. And when we look at it, if you remember, when we looked at this histologically, one of the big keys to our large intestine is it has prominent intestinal crypts. And those intestinal crypts have massive number of goblet cells in there, because while it doesn't produce any secretions like an intestinal juice or pancreatic juice, it does produce a large amount of mucus to help to grease the wheels, to facilitate the movement of that chyme into feces and then the expulsion of that feces from the body. As we talked about, there are, way back when we talked about mechanical activities, there are several specialized mechanical processes involved with the involving the large intestine. One of those is the gastroileal reflex. As we talked about, when the stomach stretches, it relaxes the ileocecal sphincter, because the goal here is the stomach is saying, hey, I've got the new load of food here, small intestine, for you to process. Whatever you've been working on, you've got to move it in on its way to the large intestine to make room for the new stuff. As we talked about, this is one of the reasons why little kids sit down, take a couple bites of food, and then have to run to the bathroom, because their gastroileal reflex sends the food into the large intestine, and that triggers our postural contractions and also the mass movements in our large intestine, and they need to defecate. That triggers defecation. Postural contractions are similar to segmentation. I'm going to cheat and go back to the picture. What happens here is a single rostrum will contract. When it contracts, it does a couple of things. It obviously is mixing and churning the chyme within it. It is compacting the chyme. This helps to draw some of the water out of it and compact it into feces. But the other thing it does is it can cause a spread of that chyme from that one hostra to the two neighboring hostras. And so when that one hostrum contracts and spreads it to the hostra, notice if it feeds it back, that isn't really helping in propulsion. So much like segmentation isn't directly related to propulsion, arguably the same thing is true for these rostral contractions. Basically, like segmentation, it is mixing and churning, so it plays a role in mixing it. Instead of this time with enzymes, it is mixing it with the bacteria that is going to help to facilitate the breakdown, rubbing it across the surface some to help in the limited absorption that takes place, but also the extraction of water and the compacting of the chyme into feces. So that's kind of what those rostral contractions do. They're compacting the chyme and absorbing the water and converting it into feces, as well as mixing it with the bacteria. These are small, minor contractions, occur about once every 30 minutes or so in kind of a wave-like fashion. But we do need propulsion within the large intestine. And we do that with what is called mass movements. Mass movements are large, powerful peristaltic waves that take place in our large intestine. On average, they occur about 3 to 4 times a day. But again, this is something that is directly related to how frequently and the volume of the food we eat. Because as we eat and our stomach stretches, remember our stomach produces gastrin. And that gastrin helps to stimulate these mass movements as well. So the more you eat, the more gastrin you're producing, the more frequent these mass movements are going to be. And their goal is moving the chyme through the large intestine and basically releasing it into that pouch-like structure that is the rectum. And by the time it reaches the rectum, it has been converted into feces. Much like the urinary system, the rectum is a pouch-like structure that accumulates the material until it is time to void, and hopefully you get to a socially appropriate location, right? Lastly, as we also talked about, our gastrocolic reflex, again, the stomach stretching not only causes the gastroileal reflex where the ileocecal valve relaxes, but it also increases the motility of the large intestine as well. Again, as new food enters the stomach, we need to move the old food out of the way to make room for it. Now, the large intestine itself doesn't do a whole heck of a lot. The only thing it releases is mucus. It does not produce any other type of intestinal juice or anything along those lines. It does not have any enzymes of its own that it produces or is bound to its surfaces. But what it does do that is very, very interesting is it holds our bacteria. We have an elaborate bacterial flora and fauna that is represented in our large intestine. And so this is where the bacteria are going to occur. And these bacteria are incredibly important and incredibly busy. They are breaking down the components of the chyme that we weren't able to break down on our own, releasing certain nutrients and other types of things so that our large intestine is able to absorb it. And so if there is a function of the large intestine, it is an absorptive function as well as propulsion, like we talked about, absorbing some water. Some of the bile salts, remember, most of the bile salts, remember, are reabsorbed in the ileum. So again, most are in the ileum, but some of the bile salts can still be reabsorbed here in the large intestine. And other organic material, and again, that organic material that is released by the bacteria. Things that we weren't able to break down on our own. One of the important things that our large intestine does is it plays an important role in the absorption of vitamins, especially lipid-soluble vitamins or vitamins that were released from bacterial activity. And B12 is one of those important ones. And again, it is here in our large intestine where that intrinsic factor that was produced by our parietal cells in the stomach play a vital role in that absorption of B12, which remember plays an important role in the formation of red blood cells. And then obviously our large intestine's job is to convert that chyme into feces, store that feces until we get to a socially appropriate location, and then expulse that feces from the body. That's about it for the large intestine. But as I mentioned, there is a tremendous amount of bacterial activity, right? And again, as we've talked about, we've mentioned this numerous times, every single person who is sitting here listening to this right now has E. coli in their body, right? Now, again, we all shouldn't be running to the hospital for that. For one thing, there's people who need it a lot more there, and nobody wants to be exposed to all those corona people. But that E. coli, as long as it's in your large intestine, is beneficial and helpful. Bacteria play an important role in breaking down the undigestible components of our food. When we ingest some of the food, some of the components are not breakdownable by our enzymes, by our structures. And so the bacteria can help in that. Bacteria help to release vitamin K, vitamin B5, as we just talked about, vitamin B12. Other electrolytes and vitamins are released that way. Now, again, the bacteria are very busy doing that. They primarily break these things down and produce their ATP anaerobically. So they're not using oxygen. They don't have mitochondria. So as a result of that, they're primarily breaking things down via glycolysis. And if you're using glycolysis, as a result of it, you get things like lactic acid and other things from that as well. Some of the bacterial byproducts that you may be familiar with is as carbohydrates are digested, it is converted into carbon dioxide and methane gas, which produces our flatus. Undigestible proteins are converted into ammonias and indoles and hydrogen sulfide gas, which provides the odor for that flatus. Right, so again, anytime you're passing that bad gas, it's not your fault, it's the bacteria, so blame your bacteria, it's not you. Of course, as we also talked about, our bilirubin that gets broken down into stercobilin and urobilin, that occurs here. And again, the urobilin, remember, is reabsorbed, released in the urine, and the stercobilin provides the color of our feces. So a lot of the components of our feces are related to that bacterial fermentation that is taking place. As I said, it enters into the large intestine as chyme, or mostly processed chyme, it's still chyme. But after somewhere between 12 and 24 hours, the breakdown and absorption is complete, the water is retracted, the feces is compacted, and the chyme is compacted and our chyme is now feces. Feces has a small component of water to it, as we've talked about. About 5% of it is the bacteria, or the products of that bacterial decomposition that we talked about, the indoles and such. And any indigestible or inorganic material. So that penny that we talked about swallowing way back in the day would come out this side, but also things that are undigestible as well. Right? When you eat a big bowl of corn with your dinner, right? The next day, you get to enjoy that corn a second time when it comes out, right? You can see the kernels in your feces. Well, not exactly. What you actually see in there, you can see bits of the corn in your feces, but really what you're seeing is the cellulose outer shell. The outer shell of the corn is made up of cellulose like grass, and cellulose we do not contain the enzymes to break down, right? A cow has, 75 stomachs that it's able to use to break it down. The sheep's the digestive system is 5.5 miles long so that it is able to deal with it. Okay, those are slight exaggerations, but those grass-eating organisms have much more elaborate digestive systems because it is so much harder to break down the cellulose. Us and our poor little bacteria are not capable of breaking down that cellulose. And so while the internal nutrients of that corn are extracted and absorbed, the outer cellulose shell remains, and that is what you actually see in your feces the next day or two after eating that corn. And then remember, as we talked about, we are constantly replacing and damaging our epithelial tissues. 2 to 4 days, you have a brand new epithelium to your small intestine. So a lot of our damaged and digested epithelial cells are released in that feces as well. All righty, questions on that? And then, like I said, the pigment. Pigment comes from the stercobilin, but again, it can also come from the foods you ingest or medications you ingest. There was a classic case, God, many years ago, of these children who were having these bright red, bright reddish pink feces. And they were doing all of these medical tests, thousands and thousands of dollars of medical tests on these kids, trying to figure out what was wrong. And part of the problem is they'd bring them into the hospital, they'd stay there for 3 days doing all these tests. All the tests would come back negative, and then it would go away. And so they'd send them home. And then a couple of days later, they'd be back in the hospital with the exact same issue again. And the short version of the story is that they were eating a special type of cereal called Frankenberry. Right, Frankenberry, it was a seasonal cereal like Count Chocula and some other one of those types of things, blueberries or whatever the other one is. And the pigment they were using in the cereal was undigestible. It was a particular red number something. I don't remember what red number it was. They've since changed it so that doesn't happen anymore. But that pigment was actually passing through and coming out in the feces. So one of the doctors finally asked the kid what they were eating. They were eating 3 bowls of this cereal every day when they got home. And so that pigment came out in their feces. Then they went to the hospital and they weren't eating it anymore. And so it went away. And so, yeah, thousands upon thousands of dollars just based on the fact that this kid was eating this crappy colored cereal. All right. Now that we've made the feces, the goal is to get it out. And to do that, once again, we are going to have a specialized structure. Again, remember, as we talked about, from the distal third of the esophagus all the way down to the rectum, right about here, remember, we have that simple columnar cells that line our digestive system. But remember, we talked about in the oral cavity and here in the anus, right, our openings to the outside world, once again, we have that non-keratinized stratified squamous epithelial tissue. And that transition point is actually located right here, called the pectinate lines. So here is that transition point between the simple columnar and the non-keratinized stratified squamous epithelial tissue. Again, as we can see, as we look at the anatomy here, starting up here at the top of the rectum, we can see there's a mucosa and our muscularis mucosa, a submucosa, 2 layers to our muscularis, right? And remember, the rectum is retroperitoneal, so there'd be an adventitia out here, and we can see some adipose anchoring it in space. Notice, again, circular and longitudinal layers of our smooth muscle. Notice there is an extension of the mucosa and submucosa, similar to what we see in the stomach, similar to what we see in the small intestine, similar to what we see in the bladder. In the stomach and in the bladder, those invaginations are rugae, and they allow for the expansion of the organ. And that might seem to make sense here in the rectum. In the small intestine, it's for surface area to increase absorption. But remember, the other thing we did is those bumps in the road slow things down. And that's probably the closest to the function of these. Inside our rectum, there are multiple, 2 or 3, of these large invaginations called the rectal valves. And really their job is to play an important role in separating the feces from the flatus. All right, so what these rectal valves allow us to do is, most of the time, successfully, we're able to expulse the flatus without expulsing feces, right? So that, when they fail, if they failed, if they weren't there, then every time you tried to pass gas, you would shart, right? So there you go. So you've got those rectal valves that help to separate the feces from the flatus. And now that I've said shart, I wonder if I have to make this, age appropriate on YouTube or something like that. Who knows? All right, so we have those. That's one of the specializations. Notice also there are these large venous structures located within the rectum. These are what are known as the hemorrhoidal veins. These, again, their goal is we're absorbing nutrients, we're absorbing those. Why these are located right by the opening is not fully understood. But what we do know is that if these veins get irritated by the feces, if the feces is too impacted, too much water, too much forcing, these can become irritated and enlarged. And of course, that is the condition we call hemorrhoids. So hemorrhoids is the irritation and enlargement of these large venous sinuses for the drainage of blood from this region. Then notice there is some similar anatomy here to what we saw in the urinary system. Notice our circular layer of the smooth muscle enlarges to form our internal anal sphincter. And again, this internal anal sphincter is comprised of smooth muscle. So it is involuntarily controlled. And it is an enlargement of the circular layer of the muscularis. But notice we also have the levator ani, which comes around, and part of the levator ani is forming our external anal sphincter. I know this doesn't quite look connected in the same way, but again, a little bit of artistic representation here. Part of that urogenital diaphragm forms an external anal sphincter, which, like in the urinary system, is skeletal muscle. And we can voluntarily control. All right. The big difference between these two is their locations. Notice here in the digestive system, the smooth internal anal sphincter is internal, external is external, and they overlap. Remember, in the urinary system, they didn't overlap. The internal was part of the bladder. The external was part of the urogenital diaphragm, so that part is the same, but they weren't located in the same location. Here, they're located the same location at the terminal part of the rectum to control this valve, the exit of the feces. And notice also, not only do we have the rectum, which has muscle to compact the valves to help to control it, but notice also within this, there are these anal columns. And these anal columns help to play a role in, again, directing and compacting the feces for expulsion out of the anus. So we did all of that. That leaves us with our defecation reflex. And our defecation reflex, if you think about it, is going to be almost identical to what we talked about in the urinary system. So we've got the pretty picture that goes over, but let's go ahead, clear this, and let's write it out again so we can see those similarities to the urinary system. It's one of those processes we learn once and we get to use. Did I take a picture of that before? I don't remember. I'll take another one just in case. learn once and use twice. So again, we need a stimulus. That stimulus is going to be contractions. Let's say it this way. We get the movement of feces into the rectum. Oops. From the sigmoid colon. And that causes a stretch of the rectum. Okay. We have stretch receptors that perceive the stretch, are stimulated. And of course, when they're stimulated, they increase their firing rate. Of action potentials. And this is going to, once again, just like we saw in the urinary system, stimulate two reflex arcs. The first is going to be local, right, or intrinsic, as we've talked about. This intrinsic local is basically going to do two main things. This is going to start contractions of the rectum, and it is going to increase contractions of the sigmoid colon. If you think about it, that is going to bring more feces to the rectum and then the expulsion of it. But we are also going to have a long or extrinsic reflex. This one involves our parasympathetic nervous system, and in particular, our sacral. It's a sacral reflex, just like urination is. And it's going to have two effects as well. The two effects of this long reflex is going to be a relaxation, the internal anal sphincter, right? But at the same time, it is going to contract the external anal sphincter. All right. So what happens here is the contractions of the rectum, the contractions of the sigmoid colon, the stretch of the rectum, right, and the relaxation of the internal anal sphincter, all are what give us that sensation that we need to void. However, thankfully, that contraction of the external anal sphincter stops us from voiding the instant you feel that sensation. So if you're sitting in your desk chair right now or in the classroom as we would normally be during this time, when you feel that need to void, you're not instantly voiding. I guess technically right now you could be sitting on the toilet listening to this lecture. It'd be very appropriate for this lecture. And so if that sensation came, out it would go, right? But the goal is to keep it in till we get to that socially appropriate location. Then, once we get to the right location, once we're ready, and we'll put that ready in quotes, then we can voluntarily release the external anal sphincter. And so when both valves are open, right, then contractions of the colon and rectum expulse the feces. Now, expulsion of feces often requires assistance. So there are some ways that we can assist that. One, as we've already talked about, is to elevate the urogenital diaphragm or more specifically, the levator ani muscle. So when we elevate that urogenital diaphragm, that we contract that levator ani, remember it elevates and can help to facilitate the release of the feces. We can also contract our abdominal muscles, and that contraction of the abdominal muscles increases the pressure in the abdominal pelvic cavity. And that increase in pressure can help to expulse the feces. But the other thing that we can do to assist this, and again, I know you've thought of it, you've done it, everyone's done it, but it is what is called the Valsalva maneuver. What we do in this case is that it's that bearing down that can occur while we're trying to defecate. Typically, the way we do this is you inflate the lungs, right? And then once you inflate the lungs, what you do is you close your glottis. And then once you close your glottis, you contract the respiratory muscles, or more specifically, contract the expiratory, which include the abdominal muscles, if you think about it. What that does is that increases, well, let's be more specific. It decreases the volume of the lungs and increases the pressure. But because your glottis is closed, the air does not leave. Instead, the air pushes down on the diaphragm. And as you push down on the diaphragm, that increases the pressure in the abdominal pelvic cavity. So that punching a grumpy face you make as you're bearing down is called the Valsalva maneuver. And that Valsalva maneuver is that increase in pressure. Now, this is all fine and good in that it helps to increase the pressure in the abdominal pelvic cavity and helps in the expulsion of the feces. But if your diet is not appropriate, if the feces is too impacted, then expulsion of the feces, defecation, can be incredibly more difficult. And one of the problems with this dramatic increase in pressure is, as we were talking about way back in 430 when we talked about the muscular system, if you increase the pressure too much in your abdominal pelvic cavity, that pressure tries to find a way out. And that can occur in the belly button region, or it can occur in the inguinal canal region. And what happens in those areas is if the pressure becomes too strong, a weak point can form, and you can get a bulging of fluid, or even in some cases organs like this or the omentum out and expressed out those conditions. And what do we call that type of condition when that occurs?
A hernia.
A hernia, either an inguinal or an umbilical hernia can occur. Now, we often think of those of that exertion of trying to lift that heavy weight or lift that car off the baby or something along those lines. But one study at one time showed that something like 40% or 30 to 40%, it's hard because not a lot of people are willing to admit this, but they speculated that as many as 30 to 40% of hernias actually occurred during defecation from trying to force the feces out too challenging. So too much pressure, too much problem, you can actually produce a hernia as a result of that. Now, one last similarity to our urinary system. As we talked about, for defecation to occur, both sphincters need to be open. And if the need to avoid occurs when you're in the car 30 minutes from the rest stop, while you might be willing to pull off to the side of the road to a bush to urinate, most people aren't willing to pull off to the side of the road to that bush to defecate. So as you continue to hold it, eventually habituation will occur. And when it occurs, the contractions stop, the internal anal sphincter contracts again. And then you finally get to the rest stop and you have difficulty voiding. But again, like in urination, as it continues to stretch, the contractions become more powerful. And if the contractions become too powerful, then the strength of those contractions can overwhelm the external anal sphincter and voiding occurs whether you want it to or not. All right. So we've done it with the pretty words. Let's do it with the pretty picture. Here we see, again, there's that stimulation. And with that stimulation, we have, again, that local reflex to stimulate the contractions. We have the long reflex to control the sphincters as a result of that. And again, it is a very similar process. Here's the pretty picture that shows this. A very similar process to what we talked about in our urination. It's both a local and parasympathetic. We get the stretch, contracts the colon walls, relaxes the internal sphincter, constricts the external one, so that we can voluntarily control it and allow voiding to occur. Like we said, right? Once both are relaxed, then defecation occurs. But if you don't relax the external, it habituates, contractions decrease. But as the continues to stretch, the reflex becomes stronger, the contractions become stronger. And again, if the pressure produced becomes greater than 55 millimeters of mercury, then it could force that external sphincter open and defecation occurs. Lastly, again, defecation is often assisted by mechanisms that help to increase the abdominal pelvic pressure, like elevating levator ani, like tensing the abdominal muscles, and like the Valsalva maneuver that we talked about. And that is your digestive system. We have now taken our mystical, magical journey from cheeseburger to poop and talked about everything else along the way. All righty. Questions on any of that? All right. With that, we are done with the digestive system. That is all the material you are going to be responsible for. We've talked histology, we've talked gross anatomy, we've talked microscopic anatomy, we've talked physiology, we've done all of those things. So you are now hopefully ready and prepared for Wednesday's exam. All righty. Any questions on any of that? All right, well then study hard. I'm going to be on my computer a lot because like I said, while I am almost done with the lab exam, there's still a couple more things I need to add. Like I said, I'm already over 200 lab exam questions. I have a few more that I need to work on and finish on. And then I'm only a little bit into your lecture exams. So I've got a lot of work. I'll be on the computer a lot, so I'll be happy and I'll be around to answer any questions you have. I do have a lecture tomorrow afternoon in my 4:30 class, so obviously I will not be available then. But most of the rest of the time, mostly because I hate my family and don't want to spend any time with them, I'm on my computer a lot, and so I should be fairly easily available. Or especially like now, because like I said, during the weekend it's a little trickier because the kids are available, and so we spend more time together. But now they're in school as well, so I've got time to work on the exam. So email me if you have any questions or concerns or comments. You have all the material to help you to be successful. Take advantage, not just of the pictures that I'm posting, but of those other resources, especially the CRC one. Their charts, all those charts are charts that are in the classroom. So when you go to that website and look at those charts, all of those charts are fair game. They're stuff that would be in the classroom, and it's going to be on there. The models are things that are in the classroom that could be available. They were lacking two important ones, so I put them on there, but that doesn't mean you shouldn't look at that stuff. You've got your PhysioX, you've got your lectures, you've got all the materials that you've used to be successful. Continue to use those, and hopefully, and obviously the lecture notes. and hopefully you can be successful on the exam. Make sure you do your histology. Well, you don't have to do it, I guess, but it's 10 points of extra credit, which is helpful, but more importantly, it's going to help you to be successful with the histology. Again, there is a lot of histology, but one of the nice things about it is all pretty distinct as long as you know where to look. The small intestine is a perfect example, right? If you just look at the 3 regions of the small intestine, it could be overwhelming because there's a lot of similarities. But as we talked about, if you know specifically where to look in the submucosa, what's going on in the submucosa instantly tells you what it is, right? So something that suddenly was challenging becomes super easy because you know where to look. And for a lot of the histology, as long as you know where to look, it hopefully will be pretty simple and straightforward. And then also, hopefully, this new format for the lab exam will limit the problems without interfering with the practicalness of the exam. So I fully admit you guys are guinea pigging this a little bit for me so that I see if there's more problems or less problems this way. I'm hoping that because it's, again, this would be even more challenging in something like the bones or the muscles where there's 85 questions on it or something like that. The fact that it's relatively small with only 65 questions, I'm hoping will limit the problems. So we'll see. Hopefully that works, and that helps you guys to be more successful with that. So we'll see how that works as well. And I'll be curious after you guys take it, what's your opinion of which one was better, because you'll have had lab exams both ways, and I'd be very curious. Maybe I'll set up a poll or something so you guys can tell me which one you liked better. All right. Be safe, study hard, email me if you have any questions. Otherwise, have a great day, and I will good luck on your exams on Wednesday. I guess I won't see you on Wednesday, but good luck on your exam on Wednesdays. I'll get to look at all your bright, shiny faces while you're taking the exams, or at least after you take the exams when I look at the videos. So, so wear a funny hat for me or something. I don't know. All right. Excellent. All right. Take care. I will talk to you guys later. Have a good day. Bye.
Thank you.